BL 4020 Special Topics, Advanced Human Endocrinology Course Syllabus, based on Williams' textbook of Endocrinology course description. This course introduces the biochemical, physiological, and clinical logic of human endocrinology. Using Williams' textbook of Endocrinology as the main reference backbone, the course examines how hormones are synthesized, secreted, transported, recognized by receptors, and integrated into feedback systems that regulate metabolism, growth, reproduction, mineral balance, and energy homeostasis. The course emphasizes a biochemistry-centered systems approach. Students will connect molecular mechanisms to organ physiology and then connect physiology to human disease. Essential question: how do hormones coordinate tissue-to-tissue communication to maintain homeostasis, regulate metabolism, and explain major endocrine disorders in humans? Instructor information. Instructor Professor Giliang Tang. Course format: lecture, guided reading, Canvas modules, and endocrine case discussion. Primary text: Williams' textbook of Endocrinology, course learning outcomes. Explain the principles of hormone synthesis, storage, release, transport, and degradation. Compare the signaling logic of peptide hormones, steroid hormones, and amine hormones. Describe how endocrine systems use negative feedback, positive feedback, and feed-forward regulation. Analyze the regulation of the hypothalamus-pituitary axes, thyroid, adrenal glands, gonads, and pancreatic islets. Explain endocrine control of food intake, body weight, glucose homeostasis, and energy balance. Interpret major endocrine diseases using mechanistic and systems-level reasoning. Course materials: Williams' textbook of Endocrinology, Canvas lecture modules and study guides, instructor lecture recordings and page audio, selected endocrine case discussions and review questions. Course organization: each module will typically include a central essential question, an introductory overview, core mechanisms and pathways, a biochem on concepts review section, a clinical connection, a section summary, 3 review questions with brief answers. Weekly course schedule. Week 1, Foundations of Endocrinology, hormone classes, receptors, signaling logic, feedback regulation, and the big picture of endocrine communication. Week 2, Hormone action and signal transduction, membrane receptors, nuclear receptors, second messengers, transcriptional effects, and endocrine responsiveness. Week 3, Hypothalamus and pituitary neuroendocrine integration, releasing hormones, pituitary control, and axis organization. Week 4, Pituitary disorders, pituitary adenomas, GH and prolactin disorders, posterior pituitary disorders, and diagnostic interpretation. Week 5, Thyroid physiology, thyroid hormone synthesis, iodide handling, thyroglobulin processing, transport, and regulation. Week 6, Thyroid disorders, hyperthyroidism, hypothyroidism, thyroiditis, nodules, and thyroid cancer. Week 7, Adrenal cortex and endocrine hypertension, steroid biosynthesis, cortisol, aldosterone, adrenal androgens, and endocrine causes of hypertension. Week 8, Reproductive endocrinology, female reproductive axis, male reproductive endocrinology, sex steroids, and fertility control. Week 9, Sexual development and function, sexual differentiation, developmental endocrine logic, and selected disorders of sex development and function. Week 10, Endocrinology in the lifespan, pregnancy, fetal development, puberty, aging, and endocrine physiology across life stages. Week 11, Mineral metabolism and bone endocrinology, calcium and phosphate regulation, parathyroid hormone, vitamin D, and metabolic bone disease. Week 12, Pancreatic endocrinology and diabetes, islet function, insulin and glucagon, glucose homeostasis, and diabetes pathophysiology. Week 13, Diabetes therapy and complications, insulin therapy, oral anti-diabetic agents, incretin-based strategies, and diabetic complications. Week 14, Appetite, obesity, and metabolic endocrinology, insulin, leptin, ghrelin, CCK, GLP-1, PYY, NPY, ARP, melanocortins, obesity, and body weight regulation. Week 15, Polyendocrine and neoplastic disorders, multiple endocrine neoplasia, polyendocrine syndromes, integrated clinical review, and final synthesis. Assessment plan: weekly Canvas reading questions or quizzes, 20%, 3 midterm exams, endocrine case discussion or presentation, 15%, final integrative exam or project, 20% expectations. Complete assigned reading before class or before opening each module. Use biochemical reasoning, not memorizational one. Participate in endocrine case analysis and discussion. Connect molecular details to physiological and clinical outcomes. Biochemi concepts essential for this course: Click to expand biochemistry concepts, protein structure and receptor function, enzyme catalysis and biosynthetic pathways, membranes and transport, signal traction including phosphorylation and second messengers, metabolic regulation and pathway integration, feedback control and homeostasis. Section summary. This course uses Williams' textbook of Endocrinology as a guiding framework while presenting endocrinology in a way that emphasizes biochemical mechanisms, organ system integration, and clinical meaning. The syllabus moves from general hormone action to specific endocrine organs and then to integrated metabolic and disease-centered modules. 3 review questions. 1. Why does this course begin with hormone action before organ systems? Because students need a mechanistic foundation before they can understand how individual endocrine organs work together. 2. Why is a systems approach important in endocrinology? Because endocrine function depends on communication among many tissues rather than isolated organ action. 3. What makes this course biochemistry-centered? It explains endocrine physiology and disease through molecular structure, signaling pathways, biosynthesis, metabolism, and regulatory logic. Back to top. Week 1. Foundations of Endocrinology. Introduction. Endocrinology is the study of how chemical signals coordinate physiology across tissues and organs. In this first module, we build the conceptual foundation for the rest of the course by asking what a hormone is, where hormones come from, how they travel, how cells detect them, and how endocrine systems use feedback to maintain homeostasis. Rather than memorizing isolated facts, this module focuses on the logic of endocrine communication. We will connect molecular structure to signaling mechanism, then connect signaling mechanism to physiological outcome. Essential question. What makes a hormone system effective at controlling human physiology across long distances and changing conditions? Learning goals for week 1. Define hormone, endocrine gland, target cell, and receptor. Classify hormones into peptide, steroid, and amine groups. Explain why some hormones act through cell surface receptors while others use intracellular receptors. Describe the role of signal transduction, amplification, and feedback regulation. Use endocrine logic to compare specificity, sensitivity, and timing of different hormonal systems. 1. What is a hormone? A hormone is a chemical messenger released by one cell or tissue that changes the behavior of another cell or tissue. In classical endocrinology, hormones are usually released into the bloodstream and act on distant targets. However, endocrine signaling exists alongside paracrine, autocrine, and neuroendocrine forms of communication. A hormone system depends on 4 linked features: a source tissue, a controlled mechanism of release, a target cell receptor, a measurable physiological response. 2. Major classes of hormones. A. Peptide and protein hormones. These hormones are made from amino acids linked into peptides or proteins. Because they are generally water-soluble, they usually circulate freely in plasma and bind to cell surface receptors. Their effects can be rapid because they trigger intracellular signaling cascades rather than diffusing across the membrane. Examples include insulin, glucagon, growth hormone, and parathyroid hormone. B. Steroid hormones. Steroid hormones are derived from cholesterol. They are relatively hydrophobic, usually travel with carrier proteins in blood, and often act through intracellular receptors that influence gene expression. Their onset is often slower than peptide signaling, but their effects may last longer. Examples include cortisol, aldosterone, testosterone, estradiol, and progesterone. C. Amine hormones. Amine hormones are derived from single amino acids, most commonly tyrosine or tryptophan. Their behavior depends on structure. For example, catecholamines are relatively water-soluble and act through membrane receptors, while thyroid hormones are hydrophobic enough to use intracellular receptor systems. 3. Why receptors matter. Hormones do not act on every cell equally. A cell responds only if it has the appropriate receptor. Receptors create specificity. They also determine the type of intracellular response, such as activation of protein kinases, generation of second messengers such as CAMP or CA2+, changes in gene transcription, changes in metabolic enzyme activity, changes in ion transport or membrane trafficking. This is why the same hormone concentration can have different effects in different tissues. 4. Cell surface receptors versus intracellular receptors. Cell surface receptors are used mainly by hormones that cannot pass easily through the lipid bilayer. These receptors convert extracellular binding into intracellular signals. This often leads to rapid responses and strong signal amplification. Intracellular receptors are used mainly by hydrophobic hormones that can cross or influence membrane passage. These receptors commonly function as transcription factors, changing which genes are expressed. This often produces slower but sustained responses. Number 5. Signal transduction and amplification. One hormone molecule binding one receptor can trigger a cascade that affects many downstream molecules. This is called signal amplification. It is a key reason why endocrine systems can produce large physiological changes even when hormone concentrations in blood are low. Important ideas include ligand binding, conformational change, second messenger formation, kinase cascades, transcriptional control. 6. Feedback regulation. Endocrine systems maintain stability through feedback loops. In negative feedback, the product or downstream effect reduces the stimulus that produced it. This stabilizes the system. In positive feedback, the response strengthens the stimulus, usually for a limited purpose such as a reproductive or developmental event. A classic endocrine logic pattern is hypothalamus, pituitary, peripheral gland, target tissues, feedback to hypothalamus and pituitary. 7. Timescale matters in endocrinology. Endocrine signals differ not only in mechanism, but also in timescale. Some hormones act within seconds or minutes, while others require hours or days to show major effects. This depends on factors such as receptor location, type of downstream signaling, need for new protein synthesis, hormone half-life in circulation, presence of binding proteins. 8. Why endocrinology is a systems science. No endocrine organ works alone. The endocrine system is a network in which the brain, pituitary, thyroid, adrenal glands, gonads, pancreas, gut, adipose tissue, liver, bone, kidney, and immune system all influence one another. Endocrinology is therefore a strong example of integrated physiology. This systems view will guide the entire course. Clinical connection. Many endocrine diseases are easiest to understand when you ask 3 questions. Is the problem one of hormone production? Is the problem one of hormone delivery or transport? Is the problem one of target cell response or feedback regulation? This framework will help later when we study disorders such as hypothyroidism, diabetes, Cushing syndrome, and hormone-resistant states. Biochem 1 concepts, essential for understanding Week 1. Click to expand. Biochem 1 concepts. Amino acids and proteins explain peptide hormone structure and receptor binding. Lipids and membranes explain why steroid hormones behave differently from peptide hormones. Protein conformation helps explain receptor activation. Enzyme regulation helps explain signaling cascades and amplification. Gene expression helps explain intracellular receptor signaling and slower hormonal effects. Homeostasis provides the logic behind endocrine feedback loops. Suggested reading guide. What features define a hormone as an endocrine signal? How do peptide, steroid, and amine hormones differ in chemistry and signaling? Why do receptors determine target specificity? How does signal amplification allow very low hormone levels to produce major effects? Why is negative feedback central to endocrine homeostasis? Section summary. Week 1 establishes the basic logic of endocrinology. Hormones are chemical messengers whose effects depend on source, transport, receptor recognition, and cellular response. Hormones can be grouped by chemistry into peptide, steroid, and amine classes, and these chemical properties strongly influence their receptor location and signaling style. Endocrine systems achieve precision through receptors, power through signal amplification, and stability through feedback regulation. This mechanistic framework will support all later modules in the course. 3 review questions. 1. Why do steroid hormones usually use intracellular receptors, while peptide hormones usually use cell surface receptors? Steroid hormones are hydrophobic and can interact with intracellular receptor systems, while peptide hormones are water-soluble and generally cannot cross the membrane easily, so they signal through surface receptors. 2. Why are receptors essential for hormone specificity? A hormone can affect only cells that express the appropriate receptor, so receptor distribution determines which tissues respond. 3. Why is negative feedback so important in endocrinology? Negative feedback stabilizes hormone systems by reducing the original stimulus once enough downstream effect has been produced. Back to top. Week 2. Hormone action and endocrine signaling. Introduction. Hormones influence physiology only when target cells can detect, transduce, and interpret their signals. This means that hormone action depends not only on hormone concentration, but also on receptor type, receptor abundance, signal transduction machinery, and the intracellular state of the target tissue. In this module, we focus on how hormones generate biological responses. We will connect chemical properties of hormones to receptor location, then connect receptors to signal cascades, gene expression, enzyme regulation, and physiological outcomes. This is the core language of endocrine communication. Essential question: how do hormones convert binding information at or within the cell into specific physiological responses? Learning goals for Week 2. Compare cell surface receptors and intracellular receptors in endocrine signaling. Explain how receptor binding initiates signal transduction. Describe major signaling strategies including G proteins, kinase cascades, second messengers, and transcriptional regulation. Explain how cells produce specific and amplified responses to hormonal signals. Discuss desensitization, downregulation, crosstalk, and hormone resistance. Hormone action begins with receptor recognition. A hormone produces an effect only when it binds an appropriate receptor. Receptors give endocrine systems their specificity. Even if two tissues are exposed to the same hormone in blood, only tissues with the correct receptor and intracellular machinery will respond strongly. Receptor-dependent hormone action involves several linked steps: hormone binding, receptor activation, signal propagation, cellular response, signal termination or adjustment. 2. Cell surface receptors. Hormones that are generally water-soluble, such as many peptide hormones and catecholamines, usually act through cell surface receptors. These hormones cannot easily cross the lipid bilayer, so information must be transmitted across the membrane by receptor proteins. Cell surface receptor signaling is often characterized by rapid onset, signal amplification, use of second messengers, reversible protein modifications such as phosphorylation. A. G protein-coupled receptors. G protein-coupled receptors, GPCRs, transmit signals by activating heterotrimeric G proteins. These G proteins then regulate downstream effectors such as adenylyl cyclase, phospholipase C, or ion channels. Many endocrine signals act through GPCR pathways. B. Receptor tyrosine kinases and related pathways. Some hormones signal through receptors with intrinsic or associated kinase activity. A key example is the insulin receptor, which activates downstream phosphorylation cascades that regulate metabolism, growth, and gene expression. C. Cytokine receptor-like signaling. Certain hormones act through receptors that recruit intracellular kinases and transcription factors. These pathways can connect receptor activation directly to changes in gene expression and are important in endocrine growth and metabolic regulation. 3. Intracellular receptors. Hormones that are sufficiently hydrophobic, such as many steroid hormones and thyroid hormones, act mainly through intracellular receptors. These receptors are often found in the cytoplasm or nucleus and commonly function as ligand-activated transcription factors. This signaling mode is often associated with slower onset than membrane signaling, longer-lasting effects, changes in gene transcription and protein synthesis, strong developmental and metabolic consequences. 4. Second messengers and signal amplification. One of the most important features of endocrine signaling is that a very small hormone signal can create a much larger intracellular response. This occurs through signal amplification. For example, one receptor activation event can generate many second messenger molecules and activate many downstream enzymes. Common signaling elements include CA2+, IP3, DAG protein kinase cascades. These signaling systems allow endocrine cells to convert weak extracellular cues into strong, coordinated intracellular actions. 5. Specificity in hormone signaling. Hormones often circulate systemically, so the body must ensure that responses remain tissue-specific. Specificity arises from several factors: which cells express the receptor, how much receptor is expressed, which signaling proteins are present downstream, whether the target cell is in a permissive or resistant physiological state, whether other hormones or nutrients are modifying the response. This is why the same hormone can produce different effects in liver, muscle, adipose tissue, bone, or brain. 6. Signal termination, desensitization, and downregulation. Endocrine signaling must be controlled not only at activation, but also at termination. If a signal persists too strongly or too long, the response can become harmful. Cells therefore use multiple mechanisms to reduce or terminate signaling. Important concepts include hormone degradation or clearance, second messenger breakdown, receptor phosphorylation and desensitization, receptor internalization, reduced receptor abundance, called downregulation. These mechanisms help explain why chronic exposure to high hormone levels can reduce sensitivity. 7. Hormone resistance. A disorder of endocrine signaling does not always mean that the hormone is absent. Sometimes the hormone is present, but the target tissue does not respond properly. This is called hormone resistance. Resistance can arise from problems at multiple levels: defective receptor structure, reduced receptor abundance, impaired intracellular signaling, altered transcriptional response, chronic nutrient or inflammatory stress. Later in the course, this concept will be especially important for understanding insulin resistance, leptin resistance, and other endocrine disorders. 8. Crosstalk among signaling pathways. Endocrine pathways do not operate in isolation. A target cell is often exposed to multiple hormones, nutrients, growth factors, and neural inputs at the same time. Therefore, hormone action depends on pathway crosstalk. One signal may amplify, weaken, or redirect the response to another. This helps explain why endocrine physiology is not simply a matter of one hormone causing one effect. Instead, physiology reflects the integration of multiple signals in real time. 9. Hormone action and timescale. Hormones differ in how fast they act and how long their effects last. Some membrane receptor pathways alter transporters or enzymes within seconds to minutes. Other hormones, especially those acting through intracellular receptors, may require hours to change gene expression and protein abundance. Timescale depends on hormone chemistry, receptor location, signaling mechanism, need for new protein synthesis, hormone clearance rate. Clinical connection. Many endocrine disorders can be understood by asking where signaling has failed. Is there too little or too much hormone? Is the receptor functioning normally? Is intracellular signal transduction intact? Has the target tissue become desensitized or resistant? This framework is useful in thinking about disorders such as diabetes mellitus, thyroid hormone resistance, as growth signaling abnormalities, and endocrine receptor mutations. Biochem I concepts, essential for understanding Week 2. Click to expand. Biochem 1 concepts. Protein structure helps explain receptor ligand specificity and conformational change. Membrane structure explains why hydrophilic and hydrophobic hormones use different receptor locations. Enzyme catalysis is essential for understanding kinase cascades and signal amplification. Phosphorylation and dephosphorylation are central regulatory mechanisms in hormone signaling. Gene expression is essential for understanding intracellular receptor action. Allostery and regulation help explain receptor activation, cooperativity, and signaling control. Suggested reading guide. Why do hydrophilic hormones usually act through membrane receptors? How do intracellular receptors differ mechanistically from membrane receptors? Why is signal amplification so important in endocrine physiology? How can the same hormone produce different effects in different tissues? Why can high hormone levels coexist with weak target cell response? Section summary. Hormone action depends on receptor recognition, signal transduction, and cellular interpretation. Water-soluble hormones generally use cell surface receptors and rapid signaling cascades, whereas hydrophobic hormones often use intracellular receptors and transcriptional control. Endocrine signaling is powerful because it is specific, amplified, and tightly regulated. Disorders can arise not only from altered hormone levels, but also from defective receptors, impaired signaling pathways, desensitization, or resistance. 3 review questions. 1. Why do peptide hormones usually act through cell surface receptors? Because peptide hormones are generally water-soluble and do not cross the lipid bilayer easily, so they transmit information through membrane receptors. 2. What is the value of signal amplification in endocrine systems? Signal amplification allows very low hormone concentrations to generate large intracellular responses through cascades of second messengers and enzymes. 3. How can hormone resistance occur even when hormone levels are high? Because the problem may lie in receptor function, receptor abundance, intracellular signaling, or target cell responsiveness, rather than in hormone production itself. Back to top. Advanced Human Endocrinology Course Design, based on Williams' textbook of Endocrinology, Introduction. This course is designed using Williams' textbook of Endocrinology as the major reference backbone. The textbook provides a strong foundation for understanding hormone synthesis, hormone secretion, hormone signaling, feedback regulation, organ-to-organ communication, and endocrine disease mechanisms. The course can be adapted for an advanced undergraduate, graduate, or medical physiology audience. It is especially suitable for a teaching style that emphasizes biochemical logic, systems integration, and clinical application. Essential question: how do hormones coordinate communication among tissues and organs to maintain homeostasis, regulate metabolism, control growth and reproduction, and explain major human endocrine disorders? Course focus. This course focuses on the biochemical and physiological principles of endocrine regulation. Students will learn how different classes of hormones act through specific receptors, how endocrine systems are organized into feedback loops, how hormones regulate energy balance, glucose homeostasis, growth, reproduction, and mineral metabolism, and how endocrine dysfunction leads to disease. Course learning outcomes. By the end of this course, students should be able to explain the major principles of hormone synthesis, secretion, transport, receptor binding, and signal transduction. Compare how the pituitary, thyroid, adrenal glands, gonads, pancreas, and bone mineral endocrine systems regulate physiology. Analyze the endocrine control of appetite, body weight, glucose metabolism, and energy homeostasis. Interpret basic endocrine laboratory patterns and relate them to disease mechanisms. Apply endocrine principles to the understanding of common human disorders such as diabetes, obesity, thyroid disease, adrenal disorders, and reproductive endocrine disorders. Recommended course model. A particularly strong version of this course is a biochemistry-centered human endocrinology course. In this design, the textbook serves as the reference backbone, while the teaching modules are organized around hormone chemistry and signaling, endocrine organ systems, metabolic regulation and fuel homeostasis, appetite, obesity and diabetes, steroid biosynthesis and reproductive endocrinology, calcium phosphate and bone regulation, clinical endocrine logic and case interpretation. Suggested 15-week course outline. Week 1, Foundations of Endocrinology. Overview of hormone classes, receptors, signal transduction, feedback regulation, and the general principles of endocrine communication. Week 2, Hormone action and endocrine signaling, membrane receptors, nuclear receptors, second messengers, transcriptional regulation, and mechanisms of endocrine responsiveness and resistance. Week 3, hypothalamus and pituitary, neuroendocrine integration, hypothalamic releasing hormones, anterior pituitary regulation, posterior pituitary function, and pituitary control of peripheral endocrine glands. Week 4, pituitary disorders, pituitary adenomas, growth hormone disorders, prolactin disorders, diabetes insipidus, SIADH, and clinical endocrine interpretation of pituitary dysfunction. Week 5, thyroid physiology, thyroid hormone synthesis, iodide metabolism, thyroglobulin processing, thyroid hormone transport, feedback regulation, and the physiological roles of T3 and T4. Week 6, thyroid disorders, hyperthyroidism, hypothyroidism, thyroiditis, goiter, thyroid nodules, and thyroid cancer, with emphasis on physiological logic and diagnosis. Week 7, adrenal cortex and steroid hormones, steroid biosynthesis, cortisol, aldosterone, adrenal androgen production, endocrine hypertension, and hormonal integration of stress and salt balance. Week 8, reproductive endocrinology, female and male reproductive axes, sex steroid action, ovarian and testicular function, reproductive cycles, fertility, and hormonal coordination of reproduction. Week 9, sex development and special topics, sexual differentiation, disorders of sex development, endocrine physiology related to gender and reproduction, and selected advanced topics in reproductive endocrinology. Week 10, endocrinology across the lifespan, fetal endocrine development, puberty, pregnancy, lactation, menopause, aging, and endocrine changes across the lifespan. Week 11, calcium phosphate and bone endocrinology, parathyroid hormone, vitamin D, calcitonin, phosphate balance, skeletal metabolism, and diseases such as osteoporosis and osteomalacia. Week 12, pancreatic endocrinology and diabetes, islet biology, insulin secretion, glucagon action, glucose homeostasis, type 1 diabetes, type 2 diabetes, and the biochemical basis of dysglycemia. Week 13, diabetes therapy and complications, insulin therapy, oral anti-diabetic drugs, incretin-based therapy, diabetic complications, and modern approaches to glucose management. Week 14, appetite, obesity, and metabolic endocrinology, endocrine regulation of food intake and body weight, including insulin, leptin, ghrelin, CCK, GLP-1, PYY, and hypothalamic neuron signals such as NPY, AGARP, and POMC melanocortins. Week 15, polyendocrine disorders and clinical integration, multiple endocrine neoplasia, polyendocrine syndromes, integrated case studies, and final synthesis of endocrine logic across organ systems. Suggested weekly module structure. Each weekly module can be organized in a consistent way for Canvas. Module title, essential question, introduction, and why it matters. Core concepts from Williams, key hormones and source tissues, target organs and signaling mechanisms, feedback loops and regulation, clinical connection or disease case. Section summary, 3 review questions with brief answers. Suggested assessment plan, weekly reading guides or short quizzes, 3 midterm quizzes or exams. Endocrine case discussion assignments, 1 hormone pathway presentation, 1 final integrative case study or final exam. Biochem 1 concepts, essential for this course. Click to expand Biochem 1 concepts, protein structure and function as the basis of receptors, transporters, and enzymes, enzyme catalysis and regulation for hormone biosynthesis and metabolic control. Membrane structure for receptor location, signaling, and transport, signal transduction including phosphorylation, second messengers, and transcriptional regulation. Metabolic pathway integration for understanding insulin, glucagon, thyroid hormone, cortisol, and leptin biology, feedback control logic for endocrine homeostasis and disease interpretation. Section summary. Williams' textbook of endocrinology can serve as a powerful backbone for building a course in human endocrinology. A strong course design begins with hormone action and feedback regulation, then expands across the major endocrine organs, and finally integrates these systems into the control of metabolism, growth, reproduction, and clinical endocrine disease. For your teaching style, the most effective model is likely a biochemistry-centered human endocrinology course that combines mechanism, systems thinking, and clinical logic. 3 review questions. 1. Why is Williams' textbook of endocrinology a strong backbone for this course? It provides a broad and integrated framework covering hormone action, organ physiology, metabolic regulation, and clinical endocrine disorders. What is the best course style for integrating your teaching interests? A biochemistry-centered endocrinology course is the best fit because it connects hormone signaling with metabolic pathways, physiological regulation, and disease mechanisms. 3. Why should weekly modules use a repeated structure? A repeated structure helps students connect core concepts, hormone mechanisms, feedback loops, and clinical applications in a consistent and organized way. Back to top. Week 3. Hypothalamus and pituitary introduction. The hypothalamus and pituitary form the central command system of endocrinology. Together, they connect the nervous system with the endocrine system, allowing information about stress, nutrient status, osmolarity, temperature, reproduction, sleep, and circadian timing to be translated into hormonal signals. In this module, we study how the hypothalamus regulates the anterior pituitary through releasing and inhibiting hormones, how the posterior pituitary releases hypothalamic neurohormones, and how the major pituitary axes control downstream endocrine glands. This module is a key bridge between signal integration and whole-body endocrine regulation. Essential question: how do the hypothalamus and pituitary gland convert neural and physiological information into coordinated endocrine control of the body? Learning goals for Week 3. Explain the anatomical and functional relationship between the hypothalamus and pituitary gland. Describe how the hypothalamus controls the anterior pituitary through releasing and inhibiting hormones. Explain how the posterior pituitary stores and releases hypothalamic hormones. Identify the major pituitary hormones and the endocrine axes they regulate. Use feedback logic to understand hypothalamic pituitary endocrine regulation. 1. Why the hypothalamus and pituitary are so important. The hypothalamus and pituitary are often described as the master regulators of the endocrine system because they coordinate multiple endocrine organs. However, it is more accurate to think of them as an integrative control center. The hypothalamus senses internal and external conditions, and the pituitary translates that information into hormone outputs that influence the thyroid, adrenal glands, gonads, mammary gland, kidney, and many other tissues. This system allows the body to coordinate stress responses, growth and development, reproduction, water balance and osmolarity, energy metabolism, circadian and seasonal timing. 2. Structural organization. The pituitary gland is functionally divided into two major parts: anterior pituitary, adenohypophysis, posterior pituitary, neurohypophysis. These two parts are connected to the hypothalamus in different ways. The anterior pituitary is regulated mainly through the hypophyseal portal blood system, whereas the posterior pituitary is directly connected to hypothalamic neurons through axons. 3. Hypothalamic control of the anterior pituitary. The hypothalamus regulates the anterior pituitary by secreting small amounts of releasing hormones and inhibiting hormones into the portal circulation. These signals travel directly to anterior pituitary cells and control the secretion of pituitary hormones. Important hypothalamic regulators include TRH, thyrotropin-releasing hormone; CRH, corticotropin-releasing hormone; GnRH, gonadotropin-releasing hormone; GHRH, growth hormone-releasing hormone; somatostatin, inhibitory regulator of several endocrine outputs, especially growth hormone; dopamine, major inhibitor of prolactin release. 4. Major hormones of the anterior pituitary. TSH, thyroid-stimulating hormone. TSH acts on the thyroid gland and promotes thyroid hormone synthesis and secretion. It is regulated primarily by TRH and negative feedback from thyroid hormones. B. ACTH, adrenocorticotropic hormone. ACTH stimulates the adrenal cortex, especially cortisol production. It is regulated mainly by CRH and by feedback from glucocorticoids. LH and FSH, luteinizing hormone LH and follicle-stimulating hormone FSH, regulate gonadal function in both females and males. They are controlled by GnRH and by feedback from sex steroids and other gonadal signals. DGH, growth hormone. GH affects growth, metabolism, and body composition. It is stimulated by GHRH, inhibited by somatostatin, and influenced by sleep, exercise, nutrition, and metabolic state. E. Prolactin. Prolactin is important in lactation and breast physiology. Unlike many pituitary hormones, its secretion is strongly restrained by hypothalamic dopamine. Thus, prolactin is notable because it is often regulated by active inhibition. Number 5. Posterior pituitary function. The posterior pituitary does not synthesize its own major hormones. Instead, it stores and releases hormones made by neurons in the hypothalamus. These hormones travel down axons into the posterior pituitary, where they are released into the bloodstream. ADH, vasopressin antidiuretic hormone, ADH, also called vasopressin, helps regulate water balance and plasma osmolarity. It promotes water reabsorption in the kidney and is strongly influenced by osmotic and volume signals. B. Oxytocin. Oxytocin is important in uterine contraction, milk ejection, and broader reproductive and social physiology. It is a classic example of neuroendocrine hormone release. 6. Major hypothalamic pituitary axes. Much of endocrinology can be organized into major regulatory axes: hypothalamus, pituitary, thyroid axis; hypothalamus, pituitary, adrenal axis; hypothalamus, pituitary, gonadal axis; hypothalamus, pituitary, growth axis; hypothalamus, posterior pituitary, kidney, uterus, mammary gland pathways. These axes make endocrine regulation easier to understand because each axis links a central signal to a peripheral response and then back to central control through feedback. 7. Negative feedback is central. The hypothalamus-pituitary system depends heavily on negative feedback. In most axes, hormones produced by peripheral endocrine glands feed back to reduce hypothalamic and pituitary stimulation. This stabilizes the system and prevents uncontrolled hormone overproduction. Examples include thyroid hormones reducing TRH and TSH drive, cortisol reducing CRH and ACH drive, sex steroids reducing hypothalamic and pituitary gonadotropin signaling in many physiological contexts, IGF-related signaling contributing to growth axis feedback. 8. Pulsatility, rhythm, and timing. Hypothalamic pituitary signaling is not always constant. Many endocrine signals are released in pulses or according to circadian rhythms. Timing matters because target tissues can respond differently to pulsatile versus continuous hormone exposure. This principle is especially important for GnRH signaling, cortisol rhythms, Growth hormone release patterns. Sleep-related endocrine regulation. 9. Neuroendocrine integration. One of the most powerful concepts in endocrinology is that the hypothalamus integrates neural information and converts it into endocrine output. Inputs related to stress, feeding, temperature, hydration, light-dark cycles, reproductive state, and emotion can all influence hypothalamic signaling. This is why the hypothalamus-pituitary system sits at the interface of brain function and hormonal control. Clinical connection disorders of the hypothalamus or pituitary can affect many endocrine systems at once. A lesion, tumor, genetic defect, or signaling abnormality at this level can produce thyroid dysfunction, adrenal dysfunction, growth abnormalities, reproductive disorders, lactation problems, water balance disorders such as diabetes insipidus. Therefore, the hypothalamus-pituitary region is central to endocrine diagnosis. When evaluating hormone disorders, clinicians often ask whether the defect is primary in the peripheral gland or secondary tertiary at the pituitary or hypothalamic level. Biochemine concepts. Peptide hormone structure is important because many hypothalamic and pituitary hormones are peptides or glycoproteins. Signal transduction is essential for understanding hypothalamic releasing hormones and pituitary responses. Membrane transport and secretion help explain neurohormone release and portal circulation logic. Feedback regulation provides the central logic of endocrine axes. Protein glycosylation and hormone heterogeneity can influence pituitary hormone behavior. Allosteric and regulatory control helps explain how multiple inputs shape hypothalamic output. Reading guide. How does the hypothalamus regulate the anterior pituitary differently from the posterior pituitary? Why is the portal circulation important for hypothalamic control of pituitary function? How do the major pituitary hormones connect to downstream endocrine glands? Why is negative feedback essential in hypothalamic pituitary axes? Why do pulsatile and circadian hormone patterns matter physiologically? Section summary. The hypothalamus and pituitary together form the central integrative hub of endocrinology. The hypothalamus receives neural and physiological information and converts it into releasing, inhibiting, or neurosecretory signals. The anterior pituitary responds through the portal system, while the posterior pituitary releases hypothalamic hormones directly into the blood. Major endocrine axes connect the hypothalamus and pituitary to downstream glands such as the thyroid, adrenal cortex, and gonads. These axes are stabilized by negative feedback and shaped by rhythmic and pulsatile hormone release. Review questions 1. What is the main functional difference between the anterior pituitary and the posterior pituitary? The anterior pituitary is regulated mainly by hypothalamic releasing and inhibiting hormones delivered through portal blood, whereas the posterior pituitary stores and releases hormones synthesized by hypothalamic neurons. 2. Why is negative feedback so important in hypothalamic pituitary endocrine axes? Negative feedback prevents overproduction of hormones and stabilizes the system by allowing peripheral hormones to reduce upstream hypothalamic and pituitary drive. 3. Why can hypothalamic or pituitary disorders affect many body systems at once? Because the hypothalamus and pituitary regulate multiple endocrine axes, a defect at this central level can alter thyroid, adrenal, gonadal, growth, lactation, and water balance physiology simultaneously. Week 4. Pituitary disorders. Introduction. Because the pituitary gland regulates multiple endocrine axes, disorders of this small organ can affect many body systems at once. A pituitary disorder may cause either excess hormone secretion, hormone deficiency, or both. In addition, a mass in the pituitary region can produce symptoms by compressing nearby structures, especially the optic chiasm. In this module, we examine the major categories of pituitary disease, including pituitary adenomas, hyperpituitarism, hypopituitarism, and posterior pituitary disorders. The goal is to understand how pituitary pathology disrupts endocrine logic and how clinicians distinguish primary, secondary, and tertiary endocrine problems. Essential question: how do disorders of the pituitary gland disrupt endocrine homeostasis, and how can we use hormone patterns and feedback logic to interpret them? Learning goals for Week 4. Classify major pituitary disorders into hormone excess, hormone deficiency, and mass effect disorders. Explain how pituitary adenomas can alter hormone secretion and produce neurological symptoms. Describe major disorders involving GH, prolactin, ACTH, and other pituitary hormones. Explain the causes and consequences of hypopituitarism. Distinguish diabetes insipidus from CH as posterior pituitary-related disorders of water balance. 1. Categories of pituitary disease. Pituitary disorders can be organized into a few major clinical categories. Hypersecretion disorders. One or more pituitary hormones are secreted in excess. Hyposecretion disorders. One or more pituitary hormones are deficient. Mass lesions. Tumors or other lesions compress nearby tissues or disrupt normal pituitary function. Posterior pituitary disorders, mainly disorders of ADH balance and water regulation. These categories may overlap. For example, a pituitary adenoma may both hypersecrete a hormone and compress normal pituitary tissue, producing deficiency in other hormones. 2. Pituitary adenomas and mass effects. Pituitary adenomas are common pituitary tumors. Some are functioning adenomas, meaning they secrete excess hormone, while others are non-functioning adenomas, meaning they mainly produce symptoms through compression. Important consequences of pituitary masses include headache, visual field defects, especially from optic chiasm compression, compression of normal pituitary tissue causing hypopituitarism, interference with hypothalamic dopamine delivery, sometimes increasing prolactin 3, prolactin disorders and prolactinomas. The most common functioning pituitary adenoma is often the prolactinoma. Excess prolactin can interfere with reproductive function because prolactin excess tends to suppress the normal hypothalamic pituitary gonadal axis. Major consequences of prolactin excess may include menstrual disturbance, infertility, galactorrhea, reduced gonadal hormone production. Prolactin levels can also rise from causes other than prolactinoma, including reduced dopamine inhibition, certain drugs, and stalk compression. 4. Growth hormone, excess and deficiency. A. Growth hormone excess. Excess growth hormone, GH, is usually caused by a GH-secreting pituitary adenoma. The clinical outcome depends on age. Before epiphyseal closure, GH excess may produce gigantism. After epiphyseal closure, it produces acromegaly, characterized by enlargement of hands, feet, jaw, and soft tissues, along with metabolic complications. B. Growth hormone deficiency. GH deficiency may arise from pituitary damage, hypothalamic disease, developmental defects, or broader hypopituitarism. In children, it can impair normal linear growth. In adults, it may contribute to altered body composition, reduced muscle mass, and decreased quality of life. 5. AH excess and Cushing disease. Excess ETH from a pituitary adenoma stimulates excess cortisol production by the adrenal cortex. When hypercortisolism is specifically driven by a pituitary ACTH-secreting adenoma, it is called Cushing disease. Cortisol excess affects many systems and may lead to central fat accumulation, muscle weakness, hyperglycemia, hypertension, skin changes, and easy bruising. This disorder is a good example of how a pituitary lesion can drive abnormal hormone production in a peripheral endocrine gland. 6. TSH and gonadotropin disorders. Less commonly, pituitary tumors or pituitary dysfunction may affect TSH, LH, and FSH. Excess TSH can drive thyroid overactivity, whereas deficiency of TSH causes secondary hypothyroidism. Deficiency of LH and FSH can impair gonadal function, fertility, and sex steroid production. These disorders remind us that pituitary disease is often best understood by asking whether downstream endocrine glands are being understimulated or overstimulated. 7. Hypopituitarism. Hypopituitarism refers to deficiency of one or more pituitary hormones. Causes include pituitary tumors, surgery, radiation, infarction, trauma, inflammatory disease, and hypothalamic disorders. The clinical effects depend on which hormones are lost. Major possibilities include adrenal insufficiency due to low ACTH, secondary hypothyroidism due to low TSH, hypogonadism due to low LH-FSH, growth problems or altered body composition due to low GH, inability to lactate due to low prolactin in certain settings. In many patients, the pattern of hormone loss provides clues about the site and severity of disease. 8. Pituitary apoplexy. Pituitary apoplexy is a medical emergency caused by hemorrhage or infarction within the pituitary, often in a pre-existing adenoma. It can present suddenly with severe headache, visual symptoms, altered consciousness, and acute endocrine failure. This condition shows how rapidly pituitary dysfunction can become life-threatening, especially when ACTH deficiency leads to acute adrenal insufficiency. 9. Posterior pituitary disorders, diabetes insipidus and SIADHA. Diabetes insipidus. Diabetes insipidus results from inadequate action of ADH. In central diabetes insipidus, there is deficient ADH production or release. This leads to impaired water reabsorption in the kidney, causing large volumes of dilute urine and increased thirst. B. SIADH syndrome of inappropriate antidiuretic hormone secretion, SIADH, involves excessive ADH effect relative to physiological need. The result is excess water retention and dilution of plasma sodium. This disorder illustrates how too much ADH can be just as dangerous as too little. 10. Primary versus secondary versus tertiary. Logic pituitary disorders are central for understanding endocrine diagnosis because they teach the difference between primary disorders, disease in the peripheral endocrine gland itself; secondary disorders, disease in the pituitary; tertiary disorders, disease in the hypothalamus. Hormone patterns can look very different depending on which level is abnormal. For example, low thyroid hormone with low or inappropriately normal TSH suggests a central problem rather than a primary thyroid problem. Clinical connection. When evaluating pituitary disease, clinicians often ask, is there evidence of hormone excess? Is there evidence of hormone deficiency? Could a mass lesion explain both the endocrine and neurological findings? Does the hormone pattern fit a primary or central endocrine disorder? This approach combines physiology, pathology, and feedback logic. Biochem on concepts, essential for understanding week 4. Click to expand, Biochem 1 concepts. Peptide hormone signaling is essential because many pituitary hormones are peptides or glycoproteins. Feedback regulation helps distinguish pituitary disease from peripheral gland disease. Signal transduction helps explain why target tissues respond abnormally when pituitary stimulation changes. Water balance and membrane transport are important for understanding ADH disorders. Protein and tissue growth regulation helps explain GH-related disorders. Metabolic integration is important because pituitary disease often affects multiple downstream organs at once. Suggested reading guide. How can a pituitary tumor cause both hormone excess and hormone deficiency? Why does prolactin excess often affect reproduction? What is the difference between gigantism and acromegaly? How do diabetes insipidus and SIADH represent opposite ADH-related disorders? Why is feedback logic so useful in diagnosing pituitary disease? Section summary. Pituitary disorders can involve hormone hypersecretion, hyposecretion, mass effects, or combinations of these. Pituitary adenomas are important because they can produce excess prolactin, GH, ACTH, or other hormones, while also compressing nearby structures and normal pituitary tissue. The module also shows why central endocrine diagnosis depends on axis logic. A pituitary disorder may alter downstream gland function even when the peripheral gland itself is normal. Posterior pituitary disorders such as diabetes insipidus and SIADH further illustrate how pituitary dysfunction can disrupt whole-body homeostasis. 3 review questions. 1. How can one pituitary lesion cause both endocrine excess and endocrine deficiency? Because a lesion such as an adenoma may hypersecrete one hormone while compressing normal pituitary tissue and reducing secretion of other hormones. 2. Why is prolactin excess often linked to reproductive dysfunction? Because prolactin excess tends to suppress normal hypothalamic pituitary gonadal signaling, which can impair gonadal hormone production and fertility. 3. What is the key physiological difference between diabetes insipidus and CAH? Diabetes insipidus reflects insufficient ADH effect and causes loss of free water, whereas SIADH reflects excessive ADH effect and causes excess water retention with dilutional hyponatremia. Back to top. Week 5. Thyroid Physiology Introduction. The thyroid gland is central to the regulation of basal metabolic rate, thermogenesis, growth, development, and the overall pace of biochemical activity in the body. Its hormones, thyroxine T4 and triiodothyronine T3, are unusual endocrine molecules because they are made from iodinated tyrosine residues within a large protein precursor called thyroglobulin. In this module, we examine how the thyroid gland concentrates iodide, synthesizes thyroid hormone, stores hormone in the follicular lumen, releases T4 and T3 into the circulation, and is controlled by the hypothalamus-pituitary-thyroid axis. Thyroid physiology is a beautiful example of endocrine biochemistry because it combines transport, oxidation chemistry, protein processing, and feedback regulation. Essential question: how does the thyroid gland synthesize, store, release, and regulate T4 and T3 to control whole-body metabolism. Learning goals for Week 5. Describe the structural organization of the thyroid follicle. Explain how the thyroid gland takes up iodide and incorporates it into hormone. Outline the steps of thyroglobulin synthesis, iodination, and coupling. Explain how T4 and T3 are released, transported, and activated in tissues. Use TRH-TSH thyroid feedback logic to understand thyroid regulation. 1. The thyroid follicle as the functional unit. The structural and functional unit of the thyroid gland is the follicle. Each follicle is lined by thyroid epithelial cells surrounding a central lumen filled with colloid. The colloid contains large amounts of thyroglobulin, which acts as both the precursor and storage matrix for thyroid hormone synthesis. This organization is unusual among endocrine glands because hormone precursor is stored outside the cell in the follicular lumen rather than entirely within cytoplasmic secretory granules. 2. Iodide uptake, the first key step. The thyroid gland must acquire iodide from the blood in order to make thyroid hormone. Thyroid follicular cells actively transport iodide across the basolateral membrane using the sodium iodide symporter. This step concentrates iodide inside the cell relative to plasma. After entering the follicular cell, iodide is moved toward the apical side and delivered into the follicular lumen, where hormone synthesis continues. Thus, thyroid hormone production depends on both transport physiology and iodine availability. 3. Thyroglobulin synthesis and secretion. Thyroglobulin is a large glycoprotein synthesized in the rough endoplasmic reticulum and Golgi apparatus of thyroid follicular cells. It contains many tyrosine residues, some of which will later become iodinated and converted into hormone-containing structures. Once synthesized, thyroglobulin is secreted across the apical membrane into the follicular lumen, where it becomes part of the colloid. At this stage, it is a protein scaffold waiting to undergo iodination and coupling chemistry. 4. Oxidation, organification, and iodination. Iodide itself cannot simply be attached directly to tyrosine without activation. At the apical surface, iodide is oxidized and used in reactions catalyzed by thyroid peroxidase, TPO. This enzyme is essential for the chemical steps that convert iodide into hormonally useful forms. The first major reaction is organification, in which iodine is incorporated into tyrosine residues within thyroglobulin. This produces T, monoiodotyrosine, DIT, diiodotyrosine. This is a distinctive biochemical strategy. The hormone precursor is built directly on tyrosine residues within a storage protein. 5. Coupling reactions form T3 and T4. After iodination, specific iodotyrosine residues within thyroglobulin undergo coupling reactions, also catalyzed by thyroid peroxidase. The two main coupling outcomes are DIT plus DIT4, MIT T3. Importantly, T3 and T4 are still attached to the thyroglobulin backbone at this point. They are formed within the protein first and released later. 6. Storage in colloid and release into blood. The thyroid gland stores substantial amounts of hormone precursor in the colloid. When stimulated by TSH, follicular cells endocytose portions of colloid containing thyroglobulin. The internalized material is then delivered to lysosomes, where proteolysis releases free T4 and T3. Free T4 and T3 leave the follicular cell and enter the bloodstream. At the same time, unused MIT and DIT can be deiodinated so that iodine can be recycled back to the thyroid cell. This recycling is an important economy mechanism in thyroid physiology. 7. T4, T3, and peripheral conversion. The thyroid gland secretes more T4 than T3, but T3 is generally the more active form at the receptor level. Many tissues convert T4 into T3 through the action of deiodinase enzymes. This means that thyroid hormone physiology is not determined only by gland secretion. It also depends on peripheral activation, inactivation, and tissue-specific hormone handling. 8. Transport in blood. Because T4 and T3 are relatively hydrophobic, most circulating thyroid hormone is bound to plasma proteins. These binding proteins help with hormone distribution and buffer rapid fluctuations in free hormone concentration. Only a small fraction of circulating hormone is free, and it is this free fraction that is most directly available for tissue action and feedback regulation. Thus, thyroid hormone physiology depends on both total hormone levels and free hormone levels. 9. Cellular action of thyroid hormone. Thyroid hormone acts mainly through intracellular receptors that regulate gene expression. By altering transcription, thyroid hormone influences many physiological processes, including basal metabolic rate, heat production, carbohydrate and lipid metabolism, growth and development, cardiovascular responsiveness, nervous system development and function. This wide range of action explains why thyroid disorders affect so many tissues and organ systems. 10. Regulation by TRH and TSH. Thyroid function is controlled by the hypothalamus-pituitary-thyroid axis. The hypothalamus secretes TRH, which stimulates pituitary release of TSH. TSH then acts on thyroid follicular cells to stimulate iodide uptake, thyroglobulin synthesis, hormone production, and hormone release. TSH is therefore not just a release signal. It supports multiple steps in thyroid structure and function, including trophic effects on the gland itself. 11. Negative feedback in the thyroid axis. Circulating thyroid hormones feed back on both the pituitary and hypothalamus to reduce further stimulation. This negative feedback is central to thyroid homeostasis. When T4 and T3 are high, TSH drive falls. When T4 and T3 are low, TSH drive rises. This feedback logic makes TSH one of the most informative markers in thyroid physiology and diagnosis. 12. Why thyroid physiology matters so much. Thyroid hormone does not simply turn one pathway on or off. Instead, it influences the overall pace and efficiency of physiology across many tissues. Its actions affect oxygen consumption, ATP turnover, nutrient metabolism, growth, and developmental timing. Because of this, even modest changes in thyroid hormone status can have broad systemic consequences. Clinical connection. Thyroid physiology provides a classic framework for interpreting endocrine laboratory patterns. Clinicians often ask, are T4 and T3 low, normal, or high? Is TSH moving in the expected opposite direction? Does the pattern suggest a primary thyroid disorder or a central disorder? Understanding the synthesis and regulation of thyroid hormone makes these diagnostic patterns much easier to understand. Biochem On concepts. Essential for understanding Week 5. Click to expand Biochem 1 concepts. Membrane transport is essential for understanding iodide uptake. Protein synthesis and glycosylation are important for thyroglobulin production. Oxidation chemistry helps explain iodide activation and iodination reactions. Tyrosine chemistry is central because thyroid hormones are made from iodinated tyrosine residues. Intracellular receptor signaling explains how thyroid hormone regulates gene expression. Feedback regulation explains TRH-TSH thyroid homeostasis. Suggested reading guide. Why is the thyroid follicle such a special endocrine structure? How do iodide uptake, iodination, and coupling lead to T3 and T4 formation? Why does the thyroid store hormone precursor in colloid? Why is TSH so important in thyroid physiology and diagnosis? Why can T4 secretion and T3 action differ across tissues? Section summary. Thyroid physiology depends on the coordinated function of the follicular cell, the colloid, and the TRH thyroid axis. The gland actively concentrates iodide, synthesizes thyroglobulin, iodinates specific tyrosine residues, couples iodotyrosenes to form T3 and T4, and stores precursor in the colloid until hormone release is needed. Circulating thyroid hormone acts broadly on metabolism, growth, and development, while negative feedback on TSH and TRH helps stabilize the system. This module provides the mechanistic foundation for understanding thyroid disorders in the next week. 3 review questions. 1. Why is thyroglobulin so important in thyroid physiology? Because thyroglobulin provides the protein scaffold on which tyrosine residues are iodinated and coupled to form T3 and T4, while also serving as a storage matrix in the colloid. 2. Why does the thyroid gland secrete more T4 than T3, yet T3 is often more biologically active? Because T4 serves largely as a circulating prohormone, while many tissues convert T4 into the more active form T3 through deiodinase enzymes. 3. Why is TSH such a valuable marker in thyroid diagnosis? Because TSH reflects pituitary response to circulating thyroid hormone through negative feedback, so it often changes predictably when thyroid hormone status changes. Week 6. Thyroid Disorders Introduction. Because thyroid hormone influences metabolism, growth, thermogenesis, cardiovascular function, and nervous system activity, disorders of the thyroid gland can affect nearly every organ system. Thyroid disease may result from abnormal hormone synthesis, autoimmune attack, inflammation, nodular growth, or neoplasia. In this module, we use feedback logic and thyroid physiology to understand major thyroid disorders, including hyperthyroidism, hypothyroidism, thyroiditis, goiter, thyroid nodules, and thyroid cancer. The goal is to understand not only what goes wrong, but also why the laboratory patterns and symptoms make mechanistic sense. Essential question: how do disorders of the thyroid gland alter T4, T3, and TSH, and how do those changes explain the clinical features of thyroid disease? Learning goals for Week 6: differentiate hyperthyroidism from hypothyroidism using physiology and hormone patterns. Explain common causes of thyroid disease, including autoimmune disorders, iodine imbalance, and nodular disease. Describe the pathophysiology of thyroiditis, goiter, and thyroid nodules. Use TSH, free T4, and related logic to distinguish primary from central thyroid disorders. Recognize the major clinical importance of thyroid cancer and structural thyroid disease. 1. A physiology-based view of thyroid disease. Thyroid disorders can be understood by asking two broad questions. Is thyroid hormone effect too high, too low, or structurally normal? Is the problem located in the thyroid gland itself, or does it arise from pituitary hypothalamic control? Using this logic, thyroid disease can be grouped into disorders of hormone excess, hormone deficiency, inflammation, enlargement, and focal lesions. 2. Hyperthyroidism and thyrotoxicosis. Hyperthyroidism refers to increased synthesis and secretion of thyroid hormone by the thyroid gland. Thyrotoxicosis refers more broadly to the clinical state caused by excess thyroid hormone action, regardless of the exact source. When thyroid hormone levels are too high, tissues are driven toward increased metabolic activity. This may cause heat intolerance, weight loss despite appetite, tachycardia and palpitations, tremor, anxiety or nervousness, increased bowel activity. These manifestations make mechanistic sense because thyroid hormone increases the pace of many metabolic and sympathetic-like responses. A. Graves' disease. A major cause of hyperthyroidism is Graves' disease, an autoimmune disorder in which antibodies stimulate the TSH receptor. This causes the thyroid gland to behave as though TSH were persistently high, even when pituitary TSH is suppressed by feedback. Graves' disease illustrates how an antibody can mimic a trophic hormone and drive gland overactivity. B. Toxic nodular disease. Hyperthyroidism can also arise from toxic multinodular goiter or a toxic adenoma, in which one or more nodules function autonomously and produce thyroid hormone without normal feedback restraint. 3. Hypothyroidism. Hypothyroidism occurs when thyroid hormone action is insufficient for the body's needs. Metabolic processes slow, and many physiological systems lose normal thyroid support. Common features may include fatigue, cold intolerance, weight gain tendency, constipation, dry skin, slowed mental or physical activity. These effects reflect reduced metabolic drive and reduced tissue responsiveness to normal physiological demands. A. Primary hypothyroidism. In primary hypothyroidism, the thyroid gland itself fails to produce adequate hormone. As a result, T4, T3 fall and TSH usually rises because feedback inhibition is reduced. B. Hashimoto thyroiditis. A major cause of primary hypothyroidism is Hashimoto thyroiditis, an autoimmune disorder in which thyroid tissue is progressively damaged by immune mechanisms. Over time, the gland loses its ability to synthesize sufficient hormone. C. Central hypothyroidism. In central hypothyroidism, the problem lies in the pituitary or hypothalamus, so TSH stimulation is inadequate. In this case, thyroid hormone is low, but TSH is not appropriately elevated. 4. Thyroiditis. Thyroiditis means inflammation of the thyroid gland. Depending on the type and stage, thyroiditis may cause temporary hormone release from damaged follicles, followed by a period of reduced hormone production. This is important because not all thyrotoxicosis comes from increased hormone synthesis. Sometimes excess hormone in blood comes from leakage of preformed hormone out of an inflamed gland. Common forms include autoimmune chronic thyroiditis, subacute inflammatory thyroiditis, postpartum thyroiditis. 5. Goiter. Goiter means enlargement of the thyroid gland. A goiter can occur in settings of normal thyroid function, hypothyroidism, hyperthyroidism. Enlargement may reflect increased TSH drive, autoimmune stimulation, iodine deficiency, or nodular remodeling. Thus, goiter is a structural description, not a complete functional diagnosis by itself. Number 6. Thyroid nodules. Thyroid nodules are common structural findings. Most are benign, but some require evaluation because a minority may represent thyroid cancer or autonomous hormone-producing tissue. From a physiology standpoint, nodules matter for two main reasons. A nodule may be non-functioning but structurally important. A nodule may be autonomously functioning and cause hormone excess. 7. Thyroid cancer. Thyroid cancer is an important structural thyroid disorder. Many thyroid cancers arise from follicular-derived cells, although they may differ greatly in behavior and prognosis. In clinical endocrinology, thyroid cancer matters because the gland is both a hormone-producing organ and a site of nodular neoplastic growth. Most thyroid cancers do not present as severe hormone excess or deficiency. Instead, they are often detected as structural lesions that require evaluation for malignant potential. 8. TSH-free T4 and diagnostic logic. Thyroid diagnosis becomes much clearer when feedback logic is used. In primary hyperthyroidism, thyroid hormone is high and TSH is usually low. In primary hypothyroidism, thyroid hormone is low and TSH is usually high. In central hypothyroidism, thyroid hormone is low, but TSH is not appropriately elevated. This pattern works because the pituitary normally adjusts TSH in response to circulating thyroid hormone. When the pattern does not follow expected feedback, a central disorder or unusual physiology should be considered. 9. Iodine balance and thyroid disease. Because the thyroid depends on iodine to synthesize hormone, iodine deficiency can contribute to hypothyroidism and goiter. Excess iodine can also disturb thyroid physiology in susceptible individuals. Thus, the thyroid gland is especially sensitive to the nutritional and environmental availability of a single trace element. Room 10. Why thyroid disorders affect the whole body. Thyroid disorders do not simply affect the neck. Because thyroid hormone helps set the pace of whole body physiology, disturbances can alter energy expenditure, body weight, cardiovascular function, gastrointestinal motility, mood, and neuromuscular performance. This explains why the symptoms of thyroid disease can seem broad but remain physiologically coherent. Clinical connection. A clinically useful way to approach thyroid disease is to ask, is the disorder mainly functional, mainly structural, or both? Do the TSH and free T4 values fit normal feedback logic? Is the problem a primary thyroid disorder, or is it central? Could inflammation, autoimmunity, or nodular autonomy explain the pattern? This approach makes thyroid diagnosis much more logical and much less dependent on memorization. Biochemical concepts feedback regulation is essential for interpreting TSH and thyroid hormone patterns. Receptor-mediated hormone action helps explain the systemic effects of excess or deficient thyroid hormone. Protein and immune interactions help explain autoimmune thyroid disease. Metabolic pathway integration explains why thyroid disorders affect weight, temperature, and energy use. Membrane transport and iodide handling remain important because iodine balance influences disease. Gene expression regulation helps explain the broad tissue effects of thyroid hormone deficiency or excess. Reading guide. Why is Graves' disease a powerful example of antibody-driven endocrine stimulation? How does primary hypothyroidism differ from central hypothyroidism in laboratory logic? Why can thyroiditis produce a temporary hyperthyroid phase? Why is goiter not the same thing as hyperthyroidism or hypothyroidism? Why can thyroid nodules be important even when hormone levels are normal? Section summary. Thyroid disorders can be grouped into problems of hormone excess, hormone deficiency, inflammation, gland enlargement, and structural lesions. Graves' disease and toxic nodular disease are important causes of hyperthyroidism, while Hashimoto thyroiditis is a major cause of hypothyroidism. Feedback interpretation using TSH and free T4 is central to diagnosis. Structural thyroid disorders such as goiter, nodules, and thyroid cancer further show that thyroid disease may involve both function and anatomy. Review questions 1. Why is Graves' disease a primary hyperthyroid disorder, even though TSH is low? Because the thyroid gland itself is overactivated by TSH receptor antibodies, while pituitary TSH is suppressed by feedback. 2. Why can thyroiditis cause transient thyrotoxicosis? Because inflamed thyroid follicles release stored thyroid hormone into the blood, even without making more hormone. 3. How do primary and central hypothyroidism differ in the lab? Primary hypothyroidism, low thyroid hormone, high TSH. Central hypothyroidism, low thyroid hormone, TSH not appropriately high. Week 14, appetite, obesity, and metabolic endocrinology, introduction. Food intake is not controlled by a single hormone or a single organ. Instead, it is regulated by an integrated network connecting the brain, gut, pancreas, adipose tissue, liver, and other metabolic organs. In this module, we examine how endocrine and neural signals work together to regulate hunger, satiety, energy expenditure, body weight, and metabolic health. This topic is a strong example of systems endocrinology. Signals from body fat, recent meals, gut nutrient sensing, and long-term metabolic state are integrated in the hypothalamus and other brain regions. Obesity develops when this system becomes chronically dysregulated, often involving insulin resistance, leptin resistance, altered gut hormone signaling, inflammation, and changes in fuel partitioning. Essential question: how do endocrine and neural signals coordinate food intake, body weight, and metabolic homeostasis? And why does this regulatory system fail in obesity? Learning goals for week 14. Explain how insulin, leptin, ghrelin, CCK, GLP-1, and PYY contribute to appetite regulation. Describe the roles of hypothalamic neuron pathways, such as NPY-ARP and POMC melanocortin signaling. Compare short-term meal signals with long-term adiposity signals. Explain why obesity is associated with insulin resistance, leptin resistance, and altered metabolic set points. Connect appetite regulation to glucose homeostasis, lipid metabolism, and metabolic disease. 1. The big picture of food intake control. Food intake is controlled by both homeostatic and hedonic systems. The homeostatic system helps maintain energy balance by detecting nutrient availability, body fat stores, and metabolic need. The hedonic system links food intake to reward, motivation, and learned behavior. In real life, eating behavior reflects the interaction of both systems. At the endocrine level, the body asks at least 3 important questions. Have we eaten recently? How much energy is stored in adipose tissue? Should fuel be stored, mobilized, or conserved? 2. Central integration in the hypothalamus. The hypothalamus is a major integration center for appetite and metabolic regulation. It receives endocrine information from blood, neural input from the gut and brainstem, and nutrient-related signals from the metabolic state of the body. Within the hypothalamus, the arcuate nucleus contains 2 functionally opposing neuron systems: ANPY-ARP neurons. NPY-ARP neurons promote food intake and are often described as hunger-promoting neurons. They are stimulated during energy deficiency and fasting. Their outputs encourage feeding and help conserve energy. B. POMC melanocortin neurons. POMC neurons generate melanocortin peptides that support satiety and increased energy expenditure. This pathway is often described as a satiety-promoting system. When activated, it opposes the feeding signals from NPY-ARP neurons. 3. Long-term adiposity signals, A. Leptin. Leptin is produced mainly by adipose tissue and reflects the level of long-term energy storage. In general, higher fat mass leads to higher leptin levels. Leptin acts on the brain to reduce hunger and support energy expenditure. It tends to suppress NP-ARP signaling and support POMC melanocortin signaling. In obesity, leptin is often high, but its effect is weaker than expected. This is called leptin resistance. The body has the signal, but the brain does not respond normally. B. Insulin. Insulin is secreted by the pancreatic base cells. It acts as both a short-term nutrient signal and a broader metabolic hormone. After meals, insulin helps promote nutrient storage, especially glucose uptake, glycogen synthesis, and lipid synthesis. It also contributes to appetite regulation in the brain. Insulin and leptin often work in the same overall direction, but they are not identical signals. Insulin is more strongly tied to recent nutrient status, while leptin reflects long-term energy stores. In obesity and type 2 diabetes, insulin resistance weakens normal metabolic control. 4. Short-term meal-related signals from the gut. A. Ghrelin. Ghrelin is often called a hunger hormone. It is produced mainly by the stomach and tends to rise before meals. Ghrelin stimulates feeding behavior and supports hunger-related signaling in the brain. B. CCK cholecystokinin, CCK, is a gut hormone released after food enters the small intestine, especially in response to fats and proteins. It promotes digestive responses and also contributes to meal termination by supporting satiety. C. GLP-1. GLP-1 is an incretin-like gut hormone that contributes to satiety, slows gastric emptying, and enhances glucose-dependent insulin secretion. It is important in both appetite regulation and glucose homeostasis. D. PYY peptide YY. PYY is released after meals and contributes to satiety. It helps reduce ongoing food intake and forms part of the gut-to-brain signaling system that tells the body that nutrients have entered the digestive tract. Number 5. Gland hormones, gut hormones, and neuron signals. One useful teaching framework is to classify appetite-regulating signals into 3 broad groups: gland or adiposity-related hormones, such as insulin and leptin; gut hormones, such as ghrelin, CCK, GLP-1, and PYY; hypothalamic neuron signals, such as NPY-AGRP and POMC melanocortins. This classification helps students distinguish where each signal comes from, what timescale it reflects, and whether it mainly promotes hunger or satiety. 6. Obesity as an endocrine and metabolic disorder. Obesity is not simply a matter of excess calorie intake in isolation. It is a chronic state involving altered endocrine signaling, fuel partitioning, inflammation, and tissue-level metabolic dysfunction. Important features include leptin resistance, insulin resistance, altered gut hormone signaling, chronic low-grade inflammation, dysregulated lipid storage and mobilization, increased risk of type 2 diabetes, fatty liver disease, and cardiometabolic disease. Thus, obesity should be understood as a disorder of metabolic regulation, not merely of body size. 7. Appetite regulation and glucose homeostasis. Appetite control is closely linked to glucose homeostasis. Hormones that affect feeding often also affect insulin secretion, insulin sensitivity, glucose utilization, and hepatic glucose production. For example, insulin lowers blood glucose and influences appetite circuits. GLP-1 supports insulin secretion and satiety. Obesity-associated insulin resistance increases the risk of dysglycemia. Chronic overnutrition shifts metabolism toward excess storage and impaired regulation. 8. Therapeutic perspective. Modern therapies for obesity and type 2 diabetes increasingly target the same pathways discussed in this module. In particular, therapies that enhance GLP-1-related signaling show how endocrine knowledge can be translated into treatment. This illustrates an important principle. Understanding endocrine pathways can directly guide rational therapy. Clinical connection. A useful clinical way to think about obesity is to ask, which satiety signals are present but not being responded to effectively? Which hunger signals are overactive or insufficiently opposed? How has altered endocrine signaling changed glucose and lipid metabolism? These questions shift the conversation from blame toward mechanism. Biochemistry concepts, essential for understanding week 14. Click to expand Biochem 1 concepts. Signal transduction explains how hormones such as insulin and leptin affect target cells and neurons. Membrane receptors are central for peptide hormones and many appetite-related signals. Metabolic pathway integration helps explain why appetite control is linked to glucose and lipid metabolism. Fuel storage and mobilization are essential for understanding obesity and insulin resistance. Homeostasis explains how endocrine systems defend energy balance and why chronic dysregulation is difficult to reverse. Feedback regulation helps explain how long-term adiposity signals and short-term meal signals interact. Suggested reading guide. What is the difference between long-term adiposity signals and short-term meal signals? How do NPY-ARP and POMC melanocortin systems oppose each other? Why are both insulin and leptin important in energy balance? Why can obesity exist even when leptin levels are high? How do appetite-regulating hormones also affect glucose metabolism? Section summary. Appetite regulation is an integrated endocrine and neural process involving gland-related hormones, gut hormones, and hypothalamic neuron signals. Signals such as insulin and leptin communicate longer-term metabolic status, while ghrelin, CCK, GLP-1, and PYY contribute to meal-related control of hunger and satiety. Obesity is best understood as a disorder of endocrine metabolic regulation, often involving leptin resistance, insulin resistance, and altered gut-brain communication. Because appetite control and glucose homeostasis are tightly linked, this topic also provides a bridge to the endocrine logic of type 2 diabetes and modern metabolic therapies. 3 review questions. 1. Why are insulin and leptin not identical appetite signals? Insulin is more closely tied to recent nutrient availability and metabolic state after meals, while leptin mainly reflects longer-term adipose energy storage. 2. What is the basic functional difference between NPY-ARP neurons and POMC melanocortin neurons? NPY-ARP neurons promote hunger and feeding, while POMC melanocortin signaling promotes satiety and tends to support energy expenditure. 3. Why should obesity be viewed as a metabolic endocrine disorder rather than only as excess body mass? Because obesity involves disrupted hormone signaling, altered fuel partitioning, insulin resistance, leptin resistance, and increased risk of broader metabolic disease. Back to top. Week 10, Endocrinology across the lifespan introduction. Endocrinology across the lifespan examines how hormone systems function differently during fetal life, infancy, childhood, puberty, adulthood, and aging. Hormones do not have identical roles at every age. Instead, endocrine priorities shift as physiology moves from growth and development to reproduction, maintenance, adaptation, and later decline or altered reserve. This module emphasizes how endocrine systems change over time, why developmental stage matters in interpreting hormone physiology, and how endocrine function reflects both timing and physiological context. The main idea is that endocrine physiology is not static, it is life stage dependent. Essential question: how do endocrine systems change from development to aging, and why must hormone physiology always be interpreted in relation to life stage? Learning goals for week 10. Explain why endocrine function differs across major stages of life. Describe key endocrine priorities in fetal life, childhood, puberty, adulthood, and aging. Connect growth, reproduction, metabolism, and aging to changing hormone physiology. Recognize why the same hormone level may have different meanings at different ages. Use lifespan thinking to interpret endocrine adaptation and vulnerability. 1. Endocrine physiology is stage-dependent. A central principle of lifespan endocrinology is that hormone function depends on developmental stage. Hormones that support organ formation in early life may later support maintenance and still later become markers of reserve, decline, or altered adaptation. Therefore, endocrine interpretation must always ask, at what stage of life is this hormone acting? Fetal and early developmental endocrinology. In fetal life, endocrine systems help coordinate organ development, growth, sex development, and preparation for birth. Hormonal signals during this period can have long-lasting effects because they shape developmental pathways rather than only short-term physiology. This stage teaches an important endocrine principle. Early hormone exposure can have organizational effects, meaning it can shape later physiology in durable ways. 3. Infancy and childhood. During infancy and childhood, endocrine priorities shift toward growth, brain development, energy balance, and maturation of organ function. Hormones such as growth hormone, thyroid hormone, insulin, and adrenal signals all contribute to healthy development. At this stage, endocrine failure often becomes visible as abnormal growth, delayed development, altered energy balance, or impaired maturation of specific tissues. Number 4. Puberty as an endocrine transition. Puberty is one of the clearest endocrine transitions in human life. During puberty, hypothalamic and pituitary signaling changes, gonadal steroid production rises, growth accelerates, and secondary sex characteristics emerge. Puberty is therefore not just a reproductive event. It is a broader endocrine reorganization involving growth, body composition, bone maturation, and reproductive capability. 5. Adulthood and endocrine maintenance. In adulthood, endocrine systems often shift from building and differentiating the body toward maintenance, reproduction, metabolic balance, and adaptation to stress. Hormone systems such as the thyroid axis, adrenal axis, reproductive axis, and pancreatic endocrine system remain active, but their goals are less about developmental construction and more about physiological stability and performance. 6. Aging and endocrine reserve. With aging, some endocrine systems change gradually in output, responsiveness, rhythmicity, or reserve. Aging does not mean that every endocrine system simply fails, but rather that endocrine physiology may show altered flexibility, recovery capacity, and set point regulation. This is why older adults may respond differently to stress, fasting, illness, temperature challenge, or metabolic imbalance than younger adults. 7. Reproductive endocrinology across age. Reproductive endocrinology provides a strong example of lifespan change. In early life, the system is organized developmentally. In puberty, it becomes activated more fully. In reproductive adulthood, it supports fertility and sex steroid physiology. Later, reproductive hormone patterns may shift again with aging, producing changes in gonadal function, fertility potential, and secondary endocrine consequences. 8. Growth, metabolism, and lifespan integration hormones involved in growth, fuel partitioning, appetite, and energy expenditure are also interpreted differently across age. A signal appropriate for rapid growth in childhood may have a different physiological meaning in adulthood or aging. This is why metabolic endocrinology and lifespan thinking are closely linked. Number 9. Hormone levels must be interpreted in context. One of the most practical principles in lifespan endocrinology is that a hormone value cannot be interpreted correctly without context. The same hormone level may be normal, abnormal, insufficient, or expected depending on age, developmental stage, physiological state, and timing. This is why endocrine reasoning always requires more than reading a number in isolation. 10. Endocrine timing matters. Lifespan endocrinology emphasizes timing. A hormone can have very different effects depending on when it acts. Developmental hormone exposure can shape tissues permanently. Pubertal activation can transform body composition and reproductive function. Adult endocrine rhythms help coordinate maintenance. Aging alters how strongly and how quickly systems respond. Thus, time is a major variable in endocrine physiology. 11. Clinical connection. A useful clinical way to think about endocrinology across the lifespan is to ask, what life stage is the patient in? What endocrine priority is dominant at that stage? Development, growth, reproduction, maintenance, or adaptation? Could a hormone pattern be normal for one age but abnormal for another? Is the endocrine system failing, adapting, or showing an age-related shift in reserve? This framework prevents oversimplified interpretation and improves physiological reasoning. 12. Endocrinology as a life course system. Endocrine physiology is best understood as a life course system. Earlier endocrine events influence later physiology. Development shapes later reserve. Puberty changes later reproductive capacity. Adult metabolic patterns affect later disease risk. Aging modifies how endocrine systems recover or compensate. This means endocrine physiology is cumulative as well as stage-specific. 13. Fast logic check. When reviewing endocrinology across the lifespan, match the stage to the dominant endocrine theme. Fetal life, organization and developmental signaling. Childhood, growth and maturation. Puberty, endocrine activation and sexual maturation. Adulthood, maintenance, reproduction, and metabolic balance. Aging, altered reserve, timing, and adaptation. This simple mapping helps students connect endocrine function to physiological context. Biochem concepts essential for understanding Week 10. Click to expand Biochem 1 concepts. Feedback regulation as a dynamic principle across age. Gene expression in developmental and steroid hormone action. Metabolic integration for growth, maintenance, and aging. Hormone signaling context as a determinant of physiological meaning. Developmental timing as a biological variable. Systems physiology linking endocrine axes across life stages. Suggested reading guide. Why must hormone physiology always be interpreted in relation to age and life stage? How do endocrine priorities differ between childhood, puberty, adulthood, and aging? Why can the same hormone concentration have different physiological meaning at different ages? How does puberty function as a major endocrine transition? Why is aging best understood as altered endocrine reserve rather than simple endocrine collapse? Section summary. Endocrinology across the lifespan emphasizes that hormone systems change with developmental stage, physiological priority, and timing. Fetal life emphasizes organization, childhood emphasizes growth, puberty emphasizes activation and maturation, adulthood emphasizes maintenance, and aging emphasizes altered reserve and adaptation. The core lesson is that endocrine physiology is not static. It must always be interpreted in context. Hormone levels, rhythms, and responses only become meaningful when linked to the stage of life in which they are acting. 3 review questions. 1. Why can the same hormone level have different meanings at different ages? Because hormone physiology depends on developmental stage, physiological priority, timing, and tissue context, rather than on concentration alone. 2. Why is puberty considered a major endocrine transition? Because puberty involves major changes in hypothalamic, pituitary, gonadal, growth, and body composition physiology rather than only a single reproductive event. 3. Why is aging better described as altered endocrine reserve than as total endocrine failure? Because many endocrine systems remain active with age, but may show altered responsiveness, timing, flexibility, and recovery capacity. Back to top. Week 12, Pancreatic Endocrinology and Diabetes Introduction. Pancreatic endocrinology is central to whole-body fuel homeostasis. The endocrine pancreas helps regulate how the body handles glucose, fatty acids, and amino acids in fed and fasting states. This system is crucial because tissues such as liver, muscle, adipose tissue, and brain all depend on coordinated endocrine signals to maintain energy balance. In this module, the main focus is the endocrine pancreas, especially insulin and glucagon, how pancreatic islet cells coordinate fuel regulation, and why diabetes mellitus represents a major disorder of endocrine metabolic control. Essential question: how does the endocrine pancreas coordinate fuel metabolism, and why does disruption of insulin-glucagon balance lead to diabetes and systemic metabolic dysfunction? Learning goals for week 12. Describe the endocrine organization of the pancreatic islet. Explain the major physiological roles of insulin and glucagon. Connect pancreatic hormone action to fed state and fasting state metabolism. Differentiate major physiological features of type 1 and type 2 diabetes mellitus. Use systems-level reasoning to understand hyperglycemia, insulin resistance, and metabolic decompensation. 1. The endocrine pancreas. The pancreas has both exocrine and endocrine functions. The endocrine component is organized into islets, which contain multiple hormone-producing cell types. These islet cells do not act independently. They form an integrated endocrine microorgan for fuel regulation. 2. Major islet hormones. The most important pancreatic endocrine hormones in metabolic homeostasis are insulin, the major anabolic glucose-lowering hormone; glucagon, the major fasting state, glucose-supporting hormone. Other islet hormones also contribute to local and systemic regulation, but insulin and glucagon provide the central logic of pancreatic endocrinology. 3. Insulin physiology. Insulin is released mainly in response to nutrient abundance, especially rising blood glucose. It promotes the uptake, storage, and use of fuels in a way that favors anabolic metabolism. In broad physiological terms, insulin promotes glucose uptake in insulin-responsive tissues, glycogen synthesis, lipid synthesis, and storage protein synthesis, and anabolic support. 4. Glucagon physiology. Glucagon becomes especially important in fasting or fuel deficit states. Its major role is to help maintain blood glucose availability, especially through actions on the liver. In broad terms, glucagon favors hepatic glycogen breakdown, hepatic glucose production, fasting state fuel mobilization. Thus, insulin and glucagon are not simply opposites in a simplistic sense. They coordinate the transition between fed and fasting metabolism. 5. Fed state and fasting state logic. Pancreatic endocrinology becomes clearest when viewed through metabolic state. Fed state, insulin influence rises, storage pathways are favored. Fasting state, glucagon influence rises, fuel mobilization and glucose support are favored by. This is why the endocrine pancreas is central to maintaining stable fuel supply despite changing nutrient availability. Verse 6. Insulin as an integrative hormone. Insulin does not act on only one tissue. It coordinates responses across liver, skeletal muscle, and adipose tissue. This makes insulin one of the strongest examples of a hormone that integrates metabolism at the whole body level. 7. Diabetes mellitus as an endocrine disease. Diabetes mellitus is not only a high glucose condition, it is a major disorder of endocrine fuel regulation. When insulin production is absent, insufficient, or ineffective relative to metabolic demand, the result is loss of coordinated nutrient handling. This means diabetes affects not only blood glucose but also lipid handling, protein balance, energy use, and long-term tissue health. 8. Type 1 diabetes mellitus. Type 1 diabetes mellitus is characterized primarily by severe loss of endogenous insulin production. The physiological result is a strong inability to maintain normal anabolic fuel regulation. Without sufficient insulin, glucose handling is impaired and fasting catabolic pathways may become exaggerated. 9. Type 2 diabetes mellitus. Type 2 diabetes mellitus is strongly associated with insulin resistance, meaning the body's tissues do not respond appropriately to insulin relative to physiological need. Over time, insulin secretion may also become unable to compensate fully for this resistance. This condition shows that hyperglycemia can emerge not only from lack of hormone, but also from impaired hormone effectiveness. Group 10. Why hyperglycemia is a systems problem. Hyperglycemia reflects more than one biochemical defect. It often involves altered liver output, altered muscle uptake, altered adipose metabolism, abnormal islet signaling, and broader endocrine or nutrient signaling imbalance. This is why pancreatic endocrinology must be understood as a systems topic rather than a single molecule topic. 11. Pancreatic endocrinology and metabolic decompensation. When pancreatic endocrine control fails severely, the body may shift into profound metabolic imbalance. This can include abnormal glucose handling, abnormal lipid mobilization, dehydration, and loss of stable metabolic coordination. These states demonstrate how central pancreatic endocrine control is to whole body physiology. 12. Clinical connection. A useful clinical way to think about pancreatic endocrinology and diabetes is to ask, is the main problem reduced insulin availability, reduced insulin effectiveness, or both? How are liver, muscle, and adipose tissue contributing to the phenotype? Is the endocrine state mainly anabolic, fasting adaptive, or pathologically unbalanced? This systems approach makes diabetes easier to understand physiologically. 13. Fast logic check. When reviewing pancreatic endocrinology, match the hormone to the main function: insulin, nutrient uptake, storage, and anabolic metabolism; glucagon, fasting support and hepatic glucose availability, fed state, insulin dominance, fasting state, glucagon dominance, diabetes, disrupted coordination of whole body fuel handling. This simple mapping helps organize the core logic of diabetes physiology. Biochem concepts, essential for understanding week 12. Click to expand Biochem 1 concepts: signal transduction for insulin receptor action, enzyme regulation for fed and fasting pathway switching, metabolic integration linking liver, muscle, and adipose tissue, membrane transport for glucose movement and tissue uptake, allostery and pathway control in glucose homeostasis, systems physiology for endocrine control of whole body fuel state. Suggested reading guide. Why are insulin and glucagon best understood together rather than separately? How does the endocrine pancreas coordinate fed state and fasting state metabolism? Why is insulin considered an integrative whole body hormone? How do type 1 and type 2 diabetes differ physiologically? Why is hyperglycemia a systems problem rather than only a glucose problem? Section summary. Pancreatic endocrinology is central to whole body fuel regulation. Insulin favors nutrient uptake and storage, while glucagon supports fasting state glucose availability. Together, these hormones help coordinate transitions between fed and fasting metabolism. Diabetes mellitus reflects failure of this coordinated endocrine system, whether through lack of insulin, insulin resistance, or both. The result is not just elevated glucose, but widespread metabolic imbalance across multiple tissues. 3 review questions. 1. Why are insulin and glucagon both required for normal metabolic homeostasis? Because the body must regulate both nutrient storage in the fed state and glucose support in the fasting state, which requires coordinated action of both hormones. 2. Why is type 2 diabetes not simply too little insulin? Because the physiology often includes insulin resistance, meaning tissues do not respond appropriately to insulin relative to metabolic need. 3. Why is diabetes considered a whole body endocrine disease? Because it alters coordinated metabolism across liver, muscle, adipose tissue, and other systems, rather than affecting glucose concentration alone. Back to top. Week 13. Diabetes therapy and complications. Introduction. Diabetes therapy is not only about lowering blood glucose. It is about restoring, as much as possible, the coordinated metabolic control that is normally provided by the endocrine pancreas and related regulatory systems. Effective therapy aims to improve glucose homeostasis, reduce metabolic stress, and prevent long-term tissue injury. This module focuses on the physiological logic behind major diabetes treatments and the mechanisms behind acute and chronic complications. The core idea is that complications arise when disordered fuel regulation persists over time and begins damaging blood vessels, nerves, kidneys, eyes, and other tissues. Essential question. How do major diabetes therapies improve metabolic control, and why does poor long-term control lead to serious complications across multiple organ systems? Learning goals for week 13. Explain the physiological goals of diabetes therapy. Differentiate major therapeutic strategies for improving glycemic control. Describe the basis of acute metabolic complications, such as severe insulin deficiency or severe hyperglycemia. Explain why chronic hyperglycemia damages blood vessels, nerves, kidneys, and eyes. Connect diabetes complications to systems-level endocrine and metabolic dysregulation. 1. The main goals of diabetes therapy. The main goals of therapy are to improve glycemic control, restore more normal fuel partitioning, reduce acute metabolic instability, prevent or slow chronic complications. This means therapy should be understood as metabolic restoration, not merely glucose suppression. 2. Lifestyle as foundational therapy. Nutritional pattern, physical activity, weight change, sleep, and broader health behaviors strongly influence glucose regulation and insulin effectiveness. This is why lifestyle therapy is foundational in diabetes care. It can improve insulin sensitivity, lower glycemic burden, and reduce pressure on pancreatic endocrine regulation. 3. Insulin therapy. Insulin therapy is essential in conditions where endogenous insulin is absent or clearly inadequate. Physiologically, insulin therapy aims to replace or supplement the missing anabolic signal that normally supports nutrient uptake, storage, and metabolic coordination. Insulin is especially central in type 1 diabetes, but may also be required in some cases of advanced type 2 diabetes when endogenous function is no longer sufficient. 4. Therapies that improve insulin effectiveness. Some diabetes therapies work primarily by improving insulin sensitivity or reducing excessive hepatic glucose output. These strategies are especially important in type 2 diabetes, where hormone resistance, rather than complete hormone absence, is often central to the physiology. The main therapeutic logic here is not replacing insulin directly, but improving how the body responds to available insulin. 5. Therapies that enhance endogenous hormone control. Some therapies improve glycemic control by enhancing endogenous pancreatic or gut pancreatic regulatory signaling. These strategies may strengthen meal-related insulin physiology, reduce inappropriate glucagon influence, or alter nutrient handling in ways that support better metabolic control. These treatments show that diabetes therapy can act through multiple endocrine pathways rather than one single mechanism. 6. Glucose monitoring and feedback. Diabetes management depends heavily on feedback-informed adjustment. Monitoring of blood glucose and related indices helps patients and clinicians assess whether therapy is restoring metabolic control or whether glycemic instability remains. This makes diabetes care a practical example of physiology-guided intervention. 7. Acute complications. Acute complications of diabetes arise when metabolic control deteriorates rapidly or severely. These states may involve extreme hyperglycemia, dehydration, abnormal lipid mobilization, altered acid-base status, and major shifts in electrolyte balance. Acute complications are especially important because they show how strongly the body depends on stable pancreatic endocrine regulation for moment-to-moment metabolic safety. Severe insulin deficiency states. When insulin deficiency is profound, the body can shift toward uncontrolled catabolic metabolism. Hepatic glucose production increases, lipid mobilization rises, and normal fuel partitioning collapses. This illustrates why insulin is necessary not only to lower glucose, but also to maintain metabolic stability. Severe hyperglycemic states. Marked hyperglycemia can produce osmotic stress, fluid loss, altered electrolyte balance, and systemic instability. These states show that glucose excess is not chemically neutral. It can reshape fluid balance and tissue function across the whole body. 8. Chronic complications. Chronic complications develop over time when hyperglycemia, metabolic stress, and related abnormalities persist. These complications are among the most important reasons why long-term control matters so much in diabetes. Chronic complications often involve small vessel damage, large vessel risk, nerve injury, kidney injury, retinal injury. 9. Microvascular complications. Long-term diabetes can damage small vessels and delicate tissues, contributing to complications involving the retina, kidney, and nervous system. These tissues are especially vulnerable because of their metabolic demands and vascular dependence. 10. Macrovascular risk. Diabetes also increases broader cardiovascular risk. This reflects not only glucose dysregulation, but also its interaction with lipid metabolism, vascular biology, inflammation-related physiology, and long-term metabolic stress. Thus, diabetes therapy must often address more than glucose alone. 11. Why complications are systems problems. Diabetes complications are not isolated events. They reflect persistent disruption of endocrine coordination, nutrient handling, vascular biology, and tissue maintenance. This is why one metabolic disorder can eventually affect eyes, kidneys, nerves, blood vessels, and healing responses at the same time. 12. Prevention as endocrine protection. Preventing complications means preserving physiological control before chronic injury accumulates. This includes better glycemic regulation, reduction of metabolic overload, improvement of hormone effectiveness, and management of associated systemic risks. In this sense, prevention is a form of endocrine protection. 13. Clinical connection. A useful clinical way to think about diabetes therapy and complications is to ask, is the main problem insufficient insulin, insulin resistance, excessive glucose production, or a mixture? Is the metabolic state acutely unstable or chronically damaging? Which tissues are most at risk from persistent poor control? This approach helps integrate therapy with complication prevention. Fast logic check. When reviewing week 13, match the concept to the main physiological meaning. Insulin therapy, restore missing anabolic endocrine signal. Insulin sensitizing therapy, improve hormone effectiveness. Acute complication, severe metabolic instability. Chronic complication, cumulative tissue injury from persistent dysregulation. Good control, complication prevention through physiological stabilization. This mapping helps organize both treatment logic and complication logic. Biochem concepts essential for understanding week 13 to expand biochem concepts, signal transduction for insulin action and resistance, fuel metabolism for fed fasting transition and decompensation, enzyme and pathway regulation for hepatic output and tissue uptake, membrane transport for glucose movement and osmotic consequences, systems physiology, linking endocrine control to tissue injury, feedback-informed treatment as applied physiology. Suggested reading guide. Why should diabetes therapy be viewed as metabolic restoration rather than only glucose lowering? How do therapies differ when the main problem is insulin absence versus insulin resistance? Why can severe insulin deficiency lead to metabolic decompensation so quickly? Why do chronic complications involve so many different tissues? How does better long-term control reduce complication risk? Section summary. Diabetes therapy aims to restore more normal metabolic coordination, whether by replacing insulin, improving insulin effectiveness, supporting endogenous regulation, or reducing metabolic stress. Therapy is best understood as an attempt to recover endocrine control of fuel handling. Complications arise when poor control persists and begins damaging multiple tissues. Acute complications reflect severe metabolic instability, while chronic complications reflect cumulative injury to vessels, nerves, kidneys, eyes, and other systems. 3 review questions. 1. Why is diabetes therapy more than just glucose lowering? Because therapy aims to restore broader metabolic coordination, not just reduce one laboratory number. 2. Why do chronic diabetes complications affect many organ systems at once? Because persistent metabolic dysregulation alters vascular biology, tissue maintenance, and cellular stress across many organs simultaneously. 3. Why is prevention such a central part of diabetes treatment? Because long-term physiological stability reduces the accumulation of tissue injury that leads to complications. Back to top. Week 11, calcium, phosphate, and bone endocrinology introduction. Calcium, phosphate, and bone physiology are tightly linked through endocrine control. These systems are essential for neuromuscular function, membrane excitability, cell signaling, mineral balance, and skeletal integrity. Bone is not simply a passive mineral storehouse. It is a dynamic tissue that participates in whole-body mineral homeostasis. In this module, the main focus is how endocrine signals maintain calcium and phosphate levels, how bone is continuously remodeled, and how organs such as the parathyroid glands, kidney, intestine, and skeleton work together as a coordinated system. Essential question: how do endocrine systems coordinate calcium, phosphate, and bone remodeling so that the body can maintain both mineral homeostasis and skeletal strength? Learning goals for week 11. Explain why calcium and phosphate must be tightly regulated. Describe the roles of PTH, vitamin D, and calcitonin in mineral homeostasis. Connect the functions of bone, kidney, intestine, and parathyroid tissue in calcium phosphate regulation. Differentiate bone formation from bone resorption and explain bone remodeling. Interpret calcium phosphate physiology using systems-level endocrine logic. 1. Why calcium and phosphate matter. Calcium is essential for muscle contraction, neurotransmitter release, intracellular signaling, secretion, and many enzyme-regulated processes. Phosphate is essential for ATP, nucleic acids, phosphorylation reactions, membranes, and mineralized tissue. Because both ions are so important, their concentrations in body fluids must be regulated within a narrow physiological range. 2. Bone is a dynamic endocrine target. Bone serves both as a structural tissue and as a mineral reservoir. However, bone is not static. It is constantly being remodeled through the coordinated activity of cells that build and cells that resorb skeletal tissue. This allows the body to maintain skeletal architecture while also adjusting mineral availability. Number 3. The main organ systems in mineral homeostasis. Calcium phosphate homeostasis depends mainly on coordination among 4 major components. Parathyroid glands sense calcium status and release PTH. Bone stores and releases mineral. Kidney regulates reabsorption, excretion, and vitamin D activation. Intestine absorbs dietary calcium and phosphate. These systems are linked by endocrine signals rather than acting independently. 4. Parathyroid hormone as a central regulator. Parathyroid hormone, PTH, is one of the main hormones regulating calcium homeostasis. It is released when blood calcium is sensed as too low. PTH acts on bone and kidney and indirectly influences intestinal absorption through vitamin D activation. In broad terms, PTH acts to increase the availability of calcium in the extracellular fluid while also shaping phosphate handling. Number 5. Vitamin D as a mineral balance hormone. Vitamin D is central to calcium and phosphate physiology because its active hormonal form supports mineral absorption and helps coordinate bone and kidney responses. The kidney is especially important because it converts precursor forms into the more active endocrine form. Vitamin D is therefore not just a nutrient topic, it is also an endocrine physiology topic. 6. Calcitonin and additional regulatory roles. Calcitonin participates in calcium physiology, although its overall role is often less dominant than PTH and vitamin D in day-to-day human mineral regulation. It is still useful conceptually because it highlights that calcium homeostasis can involve signals that oppose excessive rises in circulating calcium. 7. Bone remodeling. Bone is continuously remodeled through 2 coordinated processes: bone formation, bone resorption. Remodeling allows the skeleton to repair microscopic damage, respond to mechanical demands, and participate in mineral homeostasis. Endocrine signals influence the balance between these processes. 8. Osteoblasts, osteoclasts, and functional roles. Bone physiology is often taught through the interaction of major bone cell types. Osteoblast-related functions build bone matrix and support mineralization. Osteoclast-related functions resorb mineralized tissue. Endocrine control often influences bone indirectly by shifting the balance of signaling between these functional systems. 9. Kidney function and calcium phosphate physiology. The kidney has a major endocrine and physiological role in mineral homeostasis. It regulates how much calcium and phosphate are retained or excreted, and it also determines the activation state of vitamin D. Thus, kidney function is central to both immediate mineral balance and longer-range endocrine control. 10. Intestinal absorption and nutrient endocrine integration. The intestine provides the route by which dietary calcium and phosphate enter the body. Endocrine signals, especially active vitamin D, influence how efficiently these minerals are absorbed. This makes mineral homeostasis an excellent example of interaction between nutrition and endocrine regulation. 11. Why calcium and phosphate must be coordinated. Calcium and phosphate cannot be understood as completely separate endocrine topics because they are chemically and physiologically linked. The body must maintain enough free calcium for signaling and enough phosphate for metabolism, while also preserving appropriate skeletal mineralization. This means regulation must be coordinated rather than independent. 12. Clinical connection. A useful clinical way to think about calcium phosphate bone endocrinology is to ask, is the main problem one of circulating calcium, circulating phosphate, or bone remodeling? Is the key driver the parathyroid glands, kidney, intestine, or bone? Does the endocrine pattern suggest altered PTH physiology, altered vitamin D physiology, or remodeling imbalance? This framework helps convert a complex mineral topic into a systems-level reasoning problem. 13. Fast logic check. When reviewing this topic, match the signal to the main function. PTH, raise calcium availability and reshape renal phosphate handling. Vitamin D, support intestinal mineral absorption and coordinated mineral physiology, bone remodeling, balance structure with mineral exchange, kidney, mineral conservation, excretion, and vitamin D activation. Intestine, dietary mineral entry under endocrine influence. This simple mapping makes the overall logic of mineral endocrinology easier to organize. Biochema concepts, essential for understanding week 11. Click to expand Biochem 1 concepts. Ion balance and membrane physiology for calcium importance. Phosphorylation and ATP biology for phosphate importance. Feedback regulation for endocrine mineral sensing. Membrane transport for renal and intestinal handling. Protein and tissue structure for bone matrix physiology. Systems physiology linking bone, kidney, intestine, and parathyroid function suggested reading guide. Why must calcium be kept within such a narrow physiological range? How do PTH and vitamin D divide labor and mineral homeostasis? Why is bone remodeling essential even in adults? How does kidney physiology make mineral endocrinology possible? Why must calcium and phosphate be understood together rather than as separate ions? Section summary. Calcium phosphate and bone endocrinology is a systems topic involving the parathyroid glands, bone, kidney, and intestine. Hormones such as PTH and active vitamin D coordinate mineral balance across these tissues. The skeleton is not only structural, it is also part of mineral regulation. Bone remodeling, renal handling, intestinal absorption, and endocrine sensing together maintain both extracellular mineral homeostasis and skeletal function. 3 review questions. 1. Why is bone considered a dynamic endocrine target rather than only a structural tissue? Because bone is continuously remodeled and participates in mineral homeostasis rather than only serving as a static support structure. 2. Why must calcium and phosphate physiology be coordinated? Because both are essential for metabolism and skeletal mineralization, and regulation of one affects the physiological meaning of the other 3. Why is the kidney so important in calcium phosphate endocrinology? Because the kidney regulates mineral retention and excretion and also activates vitamin D, linking immediate handling to endocrine control. Back to top. Week 7, Adrenal Cortex and Steroid Hormones, Introduction. The adrenal cortex produces steroid hormones that regulate salt balance, blood pressure, stress responses, fuel metabolism, and part of androgen physiology. All adrenal cortical hormones come from cholesterol. This module focuses on the 3 cortical zones, the major steroid classes they make, and the main regulatory systems that control them. Essential question: how does the adrenal cortex convert cholesterol into different steroid hormones, and how do those hormones regulate volume, stress, and metabolism? Learning goals for week 7. Identify the 3 zones of the adrenal cortex. Explain why all adrenal cortical steroids begin with cholesterol. Differentiate aldosterone, cortisol, and adrenal androgens. Describe regulation by the RAS and the HPA axis. Connect adrenal physiology to blood pressure, stress, and metabolic balance. 1. Adrenal gland overview. The adrenal gland has 2 major parts: adrenal cortex, steroid hormones, adrenal medulla, catecholamines. This week focuses on the cortex. The medulla is important, but it belongs mainly to catecholamine physiology, whereas the cortex specializes in steroid biosynthesis. 2. The 3 cortical zones. A. Zona glomerulosa produces aldosterone, the main mineralocorticoid. It regulates Na+, K+, fluid volume, and blood pressure. B. Sashikal produces cortisol, the main glucocorticoid. It supports stress responses and regulates carbohydrate, lipid, and protein metabolism. C. Zona reticularis produces adrenal androgens. These contribute to the total androgen pool and are especially relevant in some developmental and clinical settings. A simple memory aid is glomerulosa, mineralocorticoids, fasciculata, glucocorticoids, reticularis, androgens. 3. Cholesterol is the starting material. All adrenal cortical steroids come from cholesterol. Cholesterol may come from circulating lipoproteins. Week 9, Sex Development and Special Topics Introduction. Sex development is a multi-step biological process involving chromosomal sex, gonadal differentiation, internal reproductive tract development, external genital development, and later pubertal maturation. Endocrine signals are central at every step. In this module, the emphasis is on how genes and hormones interact to guide development, why androgens, anti-Müllerian hormone, and gonadal differentiation signals are so important, and how variations in these pathways create clinically important developmental patterns. Essential question: how do genes, gonads, and hormones interact to direct human sex development, and why can changes in these pathways produce different developmental outcomes? Learning goals for Week 9. Differentiate chromosomal, gonadal, and phenotypic sex development. Explain how testis differentiation and ovarian differentiation influence reproductive tract development. Describe the roles of testosterone, dihydrotestosterone, and anti-Müllerian hormone. Connect sex development to puberty, secondary sex characteristics, and later endocrine physiology. Recognize why disorders or variations of sex development require pathway-based endocrine reasoning. 1. Sex development has multiple levels. Sex development is not controlled by one single switch. It involves several linked levels: chromosomal sex, gonadal sex, internal duct development, external genital development, pubertal endocrine maturation. This layered view is essential because developmental outcomes depend on the success of multiple endocrine and genetic steps, not just one hormone or one chromosome. 2. Chromosomal and gonadal differentiation. Early embryonic gonads are initially bipotential. They can develop toward testes or ovaries, depending on the genetic and signaling context. A key early developmental question is whether the gonad differentiates into a testis or an ovary, because that choice shapes later hormone production and reproductive tract development. 3. Why the testis matters early in development. Once a testis develops, it produces two especially important developmental signals: testosterone, anti-Müllerian hormone, AMH. These signals help determine whether internal and external structures follow male typical developmental pathways. Thus, early testicular endocrine output plays a major directing role in sex development. Number 4. Internal duct development. Early embryos contain ducts that can support different reproductive tract outcomes. AMH promotes regression of Müllerian structures in typical male development, while testosterone supports Wolffian duct development. If these signals are absent or altered, internal reproductive tract development follows a different pattern. Number 5. External genital development. External genital development depends strongly on androgen signaling, especially dihydrotestosterone, DHT, a more potent derivative of testosterone in specific tissues. This is why defects in androgen production, androgen conversion, or androgen receptor action can strongly influence external developmental phenotype. Number 6. Ovarian development and the absence of testicular signals. Typical ovarian development does not simply mean nothing happens. It reflects an active developmental pathway, but one not driven by the early testicular signals of testosterone and AMH. This leads to different internal and external developmental outcomes. Thus, sex development is best understood as coordinated pathway selection rather than as one pathway being merely default and passive. Number 7. Puberty as a second major endocrine stage. Sex development is not completed in embryogenesis. Puberty represents a second major endocrine phase, during which the hypothalamic pituitary gonadal axis becomes more active, gonadal steroid production rises, and secondary sex characteristics develop. Puberty therefore connects developmental endocrinology to the reproductive endocrinology studied in Week 8. Number 8. Secondary sex characteristics. Estrogens, progesterone, and androgens contribute to the development of secondary sex characteristics during puberty. These include body composition changes, breast development, body hair distribution, voice changes, and broader reproductive maturation. These effects depend on both hormone concentration and tissue responsiveness. Number 9. Androgen action and tissue conversion. One important endocrine principle is that hormone action is often tissue-specific. Testosterone can act directly in some tissues, but in others it may be converted into DHT or into estrogens. This means that hormone effects depend not only on what is secreted into blood, but also on local tissue metabolism and receptor signaling. 10. Why sex development is a systems topic. Sex development cannot be explained by a single hormone or single gene alone. It depends on coordinated success across multiple levels: gene regulation, gonadal formation, hormone synthesis, hormone conversion, receptor action, tissue response, and developmental timing. This is why developmental outcomes can differ when any one of these steps changes. 11. Special topics in sex development. Special topics in this area often include pubertal timing, androgen insensitivity, 5' reductase-related pathways, and other differences or disorders of sex development. These conditions are especially useful for teaching because they show how endocrine physiology becomes visible when one part of the system is altered. 12. Clinical connection. A useful clinical way to think about sex development is to ask, what is the chromosomal context? What is the gonadal outcome? What hormones are being produced, converted, or not responded to? At what developmental stage did the pathway change matter most? This logic helps students understand why a developmental endocrine phenotype often reflects a pathway problem rather than a simple single-label diagnosis. 13. Fast logic check. When reviewing sex development, match the signal to the main function. SRI-related signaling, testis differentiation context. AMH, Müllerian regression in typical male development, testosterone, Wolffian support, and androgen actions. DHT, external genital androgen signaling, pubertal gonadal steroids, secondary sex characteristics. This simple pathway mapping makes developmental endocrinology easier to organize. Biochemi concepts essential for understanding Week 9. Click to expand Biochemi concepts, generegulation for developmental pathway control, steroid hormone biosynthesis for testosterone and estrogen pathways, hormone conversion for testosterone to DHT to estrogen-related tissue effects, intracellular receptor signaling for sex steroid action, feedback regulation for pubertal and gonadal endocrine control, developmental timing as a principle of physiological outcome, suggested reading guide Why is Sex Development Best? Understood as a multi-step pathway rather than a single switch. How do testosterone, DHT, and AMH differ in developmental role? Why can tissue-specific hormone conversion change developmental outcome? How does puberty continue sex development rather than simply begin reproduction? Why are pathway-based explanations important in special topics of sex development? Section summary. Sex development involves multiple linked levels, including chromosomal context, gonadal differentiation, internal duct development, external development, and pubertal maturation. Hormones such as testosterone, DHT, and AMH are central signals in these pathways. This topic is best understood as a systems-level endocrine process. Developmental outcomes depend on gene regulation, hormone synthesis, hormone conversion, receptor action, and timing. That is why special topics in sex development are powerful examples of integrated endocrine reasoning. 3 review questions. 1. Why is sex development not controlled by a single hormone alone? Because sex development depends on coordinated success across genes, gonads, hormone production, hormone conversion, receptor action, and developmental timing. 2. Why is DHT especially important in some developmental contexts? Because some tissues depend strongly on the more potent androgen signal generated by conversion of testosterone to DHT. 3. Why should puberty be viewed as part of sex development rather than a completely separate topic? Because puberty is a second major endocrine stage in which gonadal steroid production rises and developmental sexual maturation becomes physiologically visible. Back to top. Week 15. Polyendocrine disorders and clinical integration. Introduction. Polyendocrine disorders remind us that endocrine physiology is not a collection of isolated glands. The endocrine system functions as an integrated network in which defects in one axis can influence other axes directly or indirectly. Some disorders affect multiple endocrine organs through autoimmunity, genetic syndromes, tumor syndromes, or broader systemic regulation. In this module, the emphasis is on how to integrate endocrine findings across multiple glands, how to think clinically when several hormone systems are abnormal at once, and why systems-level reasoning is essential for diagnosis, management, and interpretation of complex endocrine disease. Essential question: how can we use clinical integration and systems-level endocrine reasoning to understand disorders that involve multiple endocrine organs at the same time? Learning goals for Week 15. Explain what is meant by a polyendocrine disorder. Recognize why autoimmune and genetic processes can affect multiple endocrine glands. Use axis logic to interpret multi-gland endocrine abnormalities. Integrate clinical findings across thyroid, adrenal, pancreatic, gonadal, pituitary, and calcium-regulating systems. Apply systems reasoning to endocrine diagnosis and patient management. 1. What makes a disorder polyendocrine? A disorder is considered polyendocrine when more than one endocrine organ or endocrine axis is involved. The glands affected may fail, become overactive, or show mixed dysfunction. What matters is that the problem cannot be understood correctly by examining only one gland in isolation. 2. Why multiple glands may be affected together. Multiple endocrine glands may be affected together for several major reasons: autoimmune targeting of several organs, inherited syndromes affecting endocrine development or tumor risk, shared regulatory or signaling pathways, one endocrine disorder causing secondary effects in another axis. This is why endocrine interpretation often requires thinking beyond the first normal laboratory result. 3. Autoimmune polyendocrine logic. Autoimmune disease is one of the most important routes by which multiple endocrine glands can become abnormal together. For example, disorders involving the thyroid, adrenal cortex, pancreatic islets, or parathyroid glands may cluster because immune dysregulation is not limited to one target tissue. This means that once one autoimmune endocrine disorder is recognized, clinicians often remain alert for others. Number 4. Genetic and syndromic endocrine integration. Some endocrine syndromes are linked by inherited mutations that increase the likelihood of abnormalities in multiple glands. These may involve endocrine hyperplasia, endocrine tumors, altered developmental signaling, or recurring patterns of gland dysfunction. In such cases, the endocrine phenotype reflects a shared underlying program rather than unrelated gland-by-gland accidents. Number 5. Axis logic still matters. Even in complex disease, the same endocrine logic still applies. Is the problem primary, secondary, or tertiary? Is the gland underactive, overactive, or structurally abnormal? Does one axis abnormality help explain another? Clinical integration does not replace physiological logic. It extends it across multiple organs. 6. The thyroid, adrenal, pancreas example. One useful way to think about polyendocrine disease is to imagine how disorders of the thyroid, adrenal glands, and pancreatic endocrine system might intersect. Altered thyroid function changes metabolism, altered adrenal function changes stress adaptation and hemodynamics, and altered pancreatic function changes fuel regulation. Together, these can create a complicated but physiologically understandable clinical picture. 7. Pituitary integration in multi-axis disease. The pituitary gland deserves special attention because pituitary disease can affect multiple endocrine axes simultaneously. A pituitary lesion may alter thyroid, adrenal, gonadal, growth-related, and lactation-related physiology at once. Thus, one anatomical location can create the appearance of widespread multi-gland endocrine failure even when the peripheral glands themselves are structurally normal. Number 8. Calcium bone endocrine integration. Polyendocrine thinking also applies to mineral balance. A patient with endocrine abnormalities affecting parathyroid function, vitamin D physiology, renal mineral handling, or thyroid-related bone turnover may present with interconnected calcium phosphate bone findings. These are not separate problems. They reflect a shared endocrine network. Number 9. Clinical integration means pattern recognition. Clinical integration in endocrinology depends strongly on pattern recognition. The question is often not which single gland is abnormal, but rather what pattern of findings suggests a shared endocrine process. This includes combining symptoms, laboratory values, imaging, family history, timing, and progression. Number 10. Time course matters. Polyendocrine disorders may unfold over time. One endocrine diagnosis may appear first, and additional abnormalities may emerge later. This is why longitudinal clinical thinking matters. A patient's endocrine state is not always fully visible at the first presentation. 11. Treatment requires prioritization. When multiple endocrine abnormalities are present, treatment requires prioritization. Some hormone deficiencies or excess states are more immediately dangerous than others. For example, loss of stable adrenal support may be more urgent than correcting slower metabolic changes. This means endocrine treatment is not only about what is abnormal, but also about what is most urgent physiologically. Number 12. Why clinical integration is the final endocrine skill. Clinical integration is often the final major skill in endocrinology because it requires using everything learned earlier: axis logic, feedback reasoning, tissue physiology, hormone action, metabolic integration, and developmental context. A strong endocrine clinician does not merely memorize disease names. They understand how endocrine systems interact and how disorders combine. 13. Clinical connection. A useful clinical approach to polyendocrine disease is to ask, Which endocrine axes are abnormal? Is there a shared autoimmune, genetic, tumor-related, or regulatory explanation? Which problem is most urgent physiologically? Could the current diagnosis predict future endocrine problems? This framework helps turn complexity into organized clinical reasoning. 14. Fast logic check. When reviewing week 15, match the idea to the main meaning. Polyendocrine disorder, multiple endocrine organs or axes involved, autoimmune cluster, more than one gland may be immune-targeted. Pituitary lesion, one site may disrupt several peripheral axes. Clinical integration, interpret the whole endocrine pattern, not one lab alone. Treatment priority, address the most dangerous physiology first. This simple framework helps organize complex endocrine cases. Biochem concepts essential for understanding week 15. Click to expand biochem concepts, feedback regulation across multiple endocrine axes, signal integration among tissues and hormones, metabolic systems, thinking for multi-organ physiology, gene to phenotype, reasoning in inherited endocrine syndromes, immune biology and autoimmune endocrine disease, clinical prioritization as applied physiology, suggested reading guide. Why are some endocrine disorders best understood as system-wide rather than gland-specific? How can autoimmune disease produce abnormalities in several endocrine organs? Why can pituitary disease mimic multi-gland endocrine failure? Why is treatment order important in complex endocrine disease? How does clinical integration unify everything learned in endocrinology? Section summary. Polyendocrine disorders involve more than one endocrine organ or axis and often arise through autoimmune clustering, inherited syndromes, tumor-related syndromes, or multi-axis regulatory disruption. These disorders show why endocrine systems must be understood as an integrated network rather than a set of isolated glands. Clinical integration requires recognizing patterns, applying axis logic, understanding urgency, and anticipating future endocrine involvement. This is the capstone form of endocrine reasoning. 3 review questions. 1. Why can one patient have abnormalities in several endocrine glands at once? Because shared autoimmune, genetic, tumor-related, or regulatory mechanisms can affect more than one endocrine organ or axis. 2. Why does pituitary disease deserve special attention in multi-axis endocrine cases? Because one pituitary lesion can alter several peripheral endocrine axes simultaneously, creating a broad endocrine phenotype. 3. Why is clinical integration considered a final endocrine skill? Because it requires combining physiology, feedback logic, pattern recognition, and treatment prioritization across multiple systems at once. Back to top.